सर आपका आवाज नहीं आ रहा है।
बोलो। पेन में आया वो लिंक।
अभी आ रही है क्या मेरी आवाज मेम से?
हाँ सर अब आ रही है।
देख रहा हूँ
मेरी आवाज आ रही है क्या आपको?
हाँ हाँ अब आ रही सर, अब आ रही।
आया है कुछ यहाँ पे। क्लिक करना है, क्या करना है इसको?
डॉक्टर भीम आर यू हियर मी?
या सर नाउ इट्स फाइन सर।
क्लिक हियर। चलो अब मैं क्या, चलो आप इंस्टॉलेशन कर दो उसको। मैंने जैसे क्लिक कर।
सर आपकी आवाज तो आ रही थी।
Anyone can confirm? Am I audible?
नॉर्मल जैसे
सर
हम
आपकी तो आ रही है।
वो तो कर सकते हैं, वो आप समझा दीजिएगा मैडम को कैसे करना है। बाकी का मुझे अपने मोबाइल से करना होगा तो उसके लिए क्या प्रोविजन होगा, कैसे करना होगा? जैसे अभी यहाँ मैंने बोल दिया द मेल एड्रेस हैज बीन सक्सेसफुली कंफर्म।
Dr. Askar Ali, good afternoon.
ठीक है, वो मैडम को आप बता दीजिएगा कौन सी ऐप है, जो भी ऐप है आप उसका प्लीज आईडी डिटेल्स बता दीजिएगा हमको, मैडम को।
गुड आफ्टरनून डॉक्टर नरेश।
हाँ आप प्लीज एक बार मैडम को समझा दीजिएगा। जैसे अब मैंने देखिए कंफर्म वो कर दिया मेरे पास, उसके बाद से अभी कोई मेल नहीं आया है ना। उसने ये बोला है कि द कंप्लीट ऑफ इंटरनेट कनेक्शन प्रोसीजर, लॉग इन टू द ऐप यूजिंग द यूजर नेम एंड पासवर्ड एंटर्ड ड्यूरिंग द रजिस्ट्रेशन। तो रजिस्ट्रेशन करना पड़ेगा हमको, है ना? परफेक्ट, ठीक है जी, है ना। बाकी आप मैनुअली प्लीज समझा दीजिएगा मैनुअली कैसे
Welcome.
करना। हाँ बस।
You are muted actually.
कुछ वो रिमोट वगैरह भी है क्या इसमें?
Presenter.
चलिए ठीक है, ओके ओके ना, ठीक है।
It's okay.
ये बात कर रही थी मैडम से।
So now we shall start
Yeah.
our webinar with the permission of the Chair, Professor Vasudeva Ji, Vice Chancellor of Niftem. Now we are going to start our sessions. This international webinar on the topic nanobiotechnology for transforming food processing sector. Our actions are our future. This is the timely initiative of the Niftem. We can thought about the applications of the nanobiotechnology not only in the agriculture but how we can apply it in the food processing sector and how we can boost up the food processing sector by the application of the nanotechnology and biotechnological aspects. Primarily, it is the agricultural biotechnology and agricultural nanotechnology which can be applied in the agri-food sector, and after that how we can benefited the food processing industries and food processing sectors at large. So we thought that why not we can have some webinar and some discussions on this and how we can take forward this issue to the international platform. So this is part of our this World Food Day celebrations, and these celebrations are going on for all the week, and we have started this with this webinar itself from today. As tomorrow is flooding with the various seminars, various types of other programs, so we thought that we should arrange this on today itself. So we have the worthy panel with us. We have Professor T.R. Sharma ji, who is the Deputy Director General of Crop Sciences from Indian Council of Agricultural Research, New Delhi, India. As many of the participants are from the various parts of the world also. So for their knowledge, I can say that the Indian Council of Agricultural Research is the highest organization which takes care all the agricultural education, agricultural research, and significant part of the agricultural extension also in the country. And Dr. T.R. Sharma is giving his leadership as a Deputy Director General of the Crop Science. We have other eminent personality in this webinar is Professor K.S. Subramanian, who is the Director Research of Tamil Nadu Agricultural University, Coimbatore, and he is the pioneer in the nanotechnology, agri-nanotechnology, you can say, work. He did his PhD from the Canada, and after that, many crores rupees projects he handled here and generated the technology in the nanotech, and now he is giving the leadership to the Tamil Nadu Agricultural University, and he is the founder of the agri nanotechnology there also. We welcome you, sir. We have other eminent speaker, Dr. Ashwini Parikh, who is also the pioneer in the genetics and the molecular breeding aspects, and also he is heading the one important institution, National Agri-Food Biotechnology. He has informed me that he is in the meeting of the DBT. So if he will free, he will present. Otherwise, from his institute, Professor Siddhant Tiwari ji is with us, and he will present the work of the NABI. Professor Tiwari is also one of the pioneer in his field, and he is the only scientist who is involved in the CRISPR technology, and he has developed the transgenic, you can say, the banana and the rice. So we welcome you, sir. We have another very important person in this meeting is Professor Asghar Ali. Professor Asghar Ali is the a professor in the University of the Nottingham, Malaysia campus. And he was also associated earlier with the ICR system also. I know his father was in the horticulture at the CS Azad Agriculture University, Kanpur, and he also studied here. And after that, he moved to the Malaysia, and he has established the only department I can say, which is named as the Center of the Post-Harvest Biotechnology. And now he is this synonym of the nano coatings and edible coatings in the world. We also welcome you, sir. Uh, we have other very important person who is giving the leadership with the FAO, USAID, World Bank, ADB, and many other similar organizations. Dr. Patrick Webb, who worked on the future food systems, and he is giving the policy leadership to the United Nations also, and to other various organizations. And presently he is the Professor Alexander McFarland's Professor Chair at the Friedman School of Nutrition, Tufts University, US. So he will be joining short while. There is the 5:30 morning in the US, but he will join within the half an hour. So I welcome all of you, and now I request to Dr. C. Vasudevappa, who is the Vice Chancellor of National Institute of Food Technology, Entrepreneurship and Management, Kundali, Haryana, and who has given us the permission to organize this important webinar. So, and he is chairing this webinar session also. So I request him to give the presidential note as well as the welcome address from the institute. Professor Vasudeva.
अभी ना पापा हम एक प्रोग्राम अटेंड कर रहे हैं ऑनलाइन बाद में कराएंगे.
Somebody has to mute. Just inform the concern.
Yeah. I mute everyone and after that allow them.
Okay. Yeah. Good afternoon, everybody, and my best wishes on of course, tomorrow is the World Food Day. On this occasion, I think it's a great way forward, especially addressing some issues of biotechnology for transforming the food processing sector. I know this has been there in my mind. I think probably till I met Dr. Sharma in Nabi, and we were discussing, and we wanted to start a translational research facility at Niftem. Probably, I think that has taken a shape now. And today we have established a nanobiotechnology center. Of course, the process is on, the equipments are getting procured, and especially addressing issues related to the food packaging and the food processing per se. I know it's a great thing which, in fact, the even the participants number, if you look at, probably there is a lot of interest by everybody to really get into this sort of a science, which is most advanced and a sort of a cutting edge technologies are developed, which can be utilized even in food, not only in agriculture, of course, in food as well. And before I just give my welcome address, probably I think, my pranams and of course, best wishes to Dr. T.R. Sharma ji, and who has been my good friend for quite some time, and he was a director at Navi, and now he has become occupied the very coveted position of DDG Crop Science, where all this research, there is a way forward. And in he being a technologist, probably I think this is the place where he can really change the face of agricultural research even in India. And we do have with us Dr. K.S. Subramaniam, who is, of course, the Director of Research from Tamil Nadu Agricultural University. And good, he has himself started, he's a nanobiotechnologist himself, and he has started a School of Agricultural Biotechnology, which I think is a good way forward. I wish him all the best again. Of course, my friend, another friend, Dr. Ashwini Parikh was to be here, and of course, in his name, we have Siddharth Tiwari, and who is also a principal scientist in Navi, and he has, and I would say, of course, I love Navi because it's a very good institute, well nurtured and well managed institution, doing a lot of work which is more related to the food systems per se. I think that is really great. Of course, I saw Dr. Asghar Ali also, who is a professor in the University of Nottingham, Malaysia. I think that's a very good thing, and in fact, having such people will lead us to some things which are really important. I would say the recent innovations become more and more important today, and looking it in the I would say angle of food processing or value addition or food packaging per se, I think is very, very important. Of course, I also welcome Mr. Dr. Patrick Webb from Fredman School of Nutrition, and he has to join from US anyway. He may take some more time, and then probably he will be addressing all of us. And most importantly, I welcome all the delegates. I would say I'm seeing more than 120 people joining the webinar. I think it speaks of its importance. And coming to, of course, the importance of this seminar. Probably we all know. So the present day contemporary agriculture food system is constantly evolving. In fact, we know that this is because of the huge demand what we are facing towards the food security or nutritional security and things like that. And this demands new knowledge, skills, and technologies to meet our expectations. So this has been the science behind it, I would say. Of course, nano-based approaches, they promise effective solutions to cope with the multiple challenges related to global agriculture, as highlighted by the growing research on the applications of nanotechnology in the agri-food sector. I know in the ICR system already nanotechnology is being used, especially for delivery of nutrients and even to have an effective increased surface area and its efficiency and all that. Probably I think this is the coverage which gives, I would say, a huge mileage for reducing the cost of, I would say, management system. And of course, the nanoscale regime offers unique characteristics like maybe the high surface to volume ratio, which imports several novel characters to materials which cannot be absorbed as bulk nanoscale. That's where I think that this nanotechnology. In fact, I visited one institute wherein the paper dipped in a nano solution cannot be torn and which can be used for a packaging. I think this is where I think the minimum, I would say, the quantity of the material can spread over a larger area and which can be used. In fact, I am telling all this in the perspective of the food technology. This is where I think we need to really look at and evolve ourselves. I know it's not only this, even the biotechnology part has a greater relevance to see that these technologies are used in food science. Of course, the smart delivery systems, biosensors, and water management systems are being now, of course, that's what I said, designed to solve the problems faced in agro food system production. But similarly, food processing sector is being, I would say, benefited through the use of smart nano chip sensors and maybe active packaging materials, nano fortified foods, and nano nutraceuticals. I think these are all the emerging areas. Especially, I would say the fortified foods and the nutraceuticals are something which are becoming more functional foods, which are becoming more important for the human body for a better health. How do we achieve this in an effective way and deliver the nutrients? That is what is something which is very, very important. I know. So maybe there are a few emerging areas of nanotechnology, and they are helping us find out their commercial, I would say, efficacy. That is very important in the agri-food sector. One, of course, could be the nano bioprocessing for transforming the agri-food waste into more valuable biochemical product using maybe nano enzyme cocktails. I think this is one area which has great relevance today, I would say, because the waste, if you look at in food production system, is around 20 to 30%. So how do we reduce, especially in perishables? I think this is one area how we can convert them into an effective or a high value product, which is very, very important. The second, of course, could be the nano pesticides and nano fertilizers. I don't have to speak much about it. Dr. Sharmaji will speak more about it. This is another big area. That apart, nanotechnology in food transportability, traceability, and logistics. This is becoming very, very important for us. In fact, yesterday we had the PM's Gati Shakti program, which was inaugurated, where also these technologies are being highlighted. How do we achieve efficiency, especially in terms of logistics? This is something, and maybe I think multimodal connectivity using some of these advanced technologies. This is one area we need to look at very seriously. I know Sharmaji will be much aware of these things, and I would say this is going to help us in a very big way to address the issues related to the food, especially the processing or a value addition or a packaging or even transportation. Of course, food forensics, I know this is a new science. Slowly and steadily, of course, now the forensic science is getting developed. Food forensics is again another branch of forensic science where we need to really look at how actually we can really bring this into the system. And of course, Internet of Nano Things. The Internet of Things is getting modified into everything. And without Internet, probably IoT, probably no science exists today. In fact, this also is another, especially the telecommunication revolution has brought in all these things. We need to really look at how best we can use this in another food science or food processing or food packaging and things like that. Despite great potential, of course, in nanotechnology, still, I would say we are bound by many regulations, and also probably we are not very sure the efficacy of these products, especially when we use in food science, especially in food processing or food products. This is one area which requires a huge attention by all the nanobiotechnologies. Probably look at the efficacy and the feasibility and the I would say sort of safety which is involved as far as the food is concerned. I know because we are dealing with a food which is very important for every human being. So we need to look at this very seriously, of course. To handle all such of these things, of course, very recently, the guidelines for evaluation of nano-based agri-input and food products in India have been developed by the Department of Science and Technology, of course, by the Department of Biotechnology. I would say these are things which need to be really seriously understood by all Indians who are into this sort of innovation. This is very, very important. Of course, Niftem also has provided its inputs, especially playing as a sort of a key, key role in while formulating these guidelines. But all the same, science is beyond everybody. We need to look at that, probably try to understand and address some of these issues. I wish these guidelines will encourage Indian innovators and, of course, industries to develop and commercialize new nano-based agri-input and food products. This is very, very important. Of course, these guidelines will pave way for the significant benefits through cutting-edge technologies for various national missions. Could be doubling the mission on doubling the farmers' income, or even the national mission on sustainable agriculture, things like that, which I feel are becoming very, very important. I know, of course, I am not a real nanobiotechnologist, but all the same, I appreciate and really admire the science which is involved, especially in addressing some of these issues at a little bit higher level, which can really help us for a better preservation of the food, processing facility, or even packaging and the shelf life of the food and things like that, which becomes very, very important. Once again, I wish the entire program a good success, and with all the inputs of very learned luminaries, probably I think it's going to be much more interesting and probably pave way for the young and the budding innovators to look into this science in a holistic approach so that they can achieve something which is useful for the society. With this, all the best once again. Thank you very much. Thanks for all of you for being with us and participating in this program. Thank you very much.
Thank you very much, sir, for giving the overview on the nanobiotechnology aspects applied in the food, and welcoming all our guests and speakers here. At present, I can see that we have more than 15 countries participants here, right from Romania to Spain to UK, Morocco, US, and some African countries also. So I welcome all the participants also here in this August gathering. We have Dr. Tilakraj Sharmaji, who is holding the the position of the Deputy Director General Crop Science in Indian Council of Agricultural Research from 2020. He was served as Project Director, ICR National Research Center on Plant Biotechnology, IRI New Delhi for 3 to 4 years, Executive Director at National Agri-Food Biotechnology Institute, Mohali for 3 years, and CEO of the Center of Innovative and Applied Bioprocessing, that is known as the CIAP at Mohali itself during this period. DBT Overseas Associate 1994 to 96. Then he was the University of Alberta, Edmonton, Canada. He was visiting scientist at Genome Research Center, Cold Spring Harbor Laboratory, New York, US in 2001, and visiting scientist at CSHL, US 2004. He has been selected as Fellow of Indian National Science Academy, National Academy of Sciences India, and National Academy of Agricultural Sciences. So all related 3 highest academies has inducted him as a fellow. His research areas are plant disease resistance, genomics, and bioinformatics. He was awarded with the University Gold Medal and Certificate of Honor in PhD, ICR Young Scientist Award in Crop Science in 1999, Professor M.J. Narasimhan Academic Merit Merit Award IPS 1999, ISCA Professor Umakant Sinha Memorial Award 2001, International World Technology Award for Biotechnology, jointly to International Rice Genome Sequencing Team, the World Technology Network 2003, DBT National Bioscience Award 2007, Vasvik Award 2011, Rafi Ahmed Kidwai Award ICR 2012, NAS Reliance Industries Platinum Jubilee Award 2013, and DST JC Bose National Fellowship 2013 and 2019, and Professor K.S. Bilgrami Memorial Medal INSA 2018. So he is giving the leadership to the country in the field of the biotechnology, in the field of the crop science research overall. And he accepted our invitation just only on the WhatsApp message and just on one email. It's the kindness of Dr. T.R. Sharmaji that he is here with us. We welcome you, sir, and we request you to provide the inaugural address this meeting. Professor Sharma. Sir, unmute, please.
Okay, thank you, Dr. Sunil Parikh, Honorable Vice Chancellor of Niftem, Professor C. Vasudevappa, distinguished participants in this webinar and speaker in this particular session, Professor K.S. Subramanian, Director Research, DNAU Coimbatore, my colleague, former colleague, Dr. Siddharth Tiwari from National Agri-Food Biotechnology Institute, Mohali, Dr. Asghar Ali from Malaysia, and distinguished participants, ladies and gentlemen. First of all, I would like to thank Professor C. Vasudevappa for and also Dr. Sunil Parikh for inviting me to this international webinar on nanobiotechnology for transforming food processing sector. Our actions are our future. This is a very topical subject on which this webinar has been now organized by Niftem, which is one of the unique institutes of the country. Before I speak few things about the introductory remarks and my talk, I would like to extend my heartiest best wishes and happy Vijayadashmi to all of you. And also, I would like to extend my best wishes to all of you on World Food Day, which is being celebrated on 16th October to 16th October. And I would like to inform everybody sitting here that in ICAR, we are going to organize World Food Day in the form of an evening with World Food Laureate from 5 p.m. to 7 p.m. And I will share the link with Dr. Sunil Parikh so that he can further share with all the participants to participate in that. So during these 10-15 minutes, which are allotted to me, I would like to tell few things about Indian agriculture. And as told by Dr. Vasudevappa, Indian Council of Agriculture Research is one of the huge establishment of the country with more than 8,000 scientists. And the division in which I am working is Crop Science Division as Deputy Director General Crops, and which leads 28 institutes and 21 All India Coordinator Research Projects. We have 3,570 scientists in Crop Science Division spread across the country, dealing with 85 crops. So you can make out now that whatever we are eating, any crops we are dealing, that is being taken care in terms of its improvement by my division in Indian Council of Agriculture Research. And I would like to give you a glimpse of the growth of food production in India after independence. We all know that before independence and before 1950, we were surviving on the food which was imported from United States of America through PL40 scheme. And we were just surviving from ship to mouth condition was there. But if you compare the food production situation from 2050 to till today, it has now increased tremendously, and we have seen green revolution in 60s and 70s. And today, if I give you a statistics of food grain production in the country, which was 50 million tons in 1950-51, has now increased to 308 million tons. That is 6 times increase from 1950. It happened all because of the new varieties and technologies developed by agricultural scientists, policy of the government, and also the hard work of farming community, which are playing very important role in this endeavor. Similarly, pulses, which was a very difficult situation for the country, and we had 8.4 metric ton in 1950, and till 2014 we were importing a lot of pulses. But now the pulse production has gone to 25 metric tons. That is around 3 times increase. Oil seeds, although the production was 5.16 metric ton, has gone to 36 metric ton in 2020-21, that is around 7% increase, but still we are importing edible oil to the tune of more than 70,000 per annum from different countries. So there is large scope in oilseed edible oil production. In case of cotton, our production has gone from 0.5 million tons to 6.2 million tons bales, and it has grown more than 11.9 times. That is all because of transgenic cotton which we are growing in the country and more improved agriculture technology. In sugarcane, our sugarcane production was 57 metric ton in 2050, has now increased to 399.25 metric ton, which is around 7 times growth. Similarly, horticulture produce, which was 96 metric ton in 2050, has now grown to 329 metric ton and around 4 times growth in this particular sector. Milk production has seen a tremendous growth under white revolution, and which was 17 million tons in 2050, has now increased to 198 metric ton, which is around 12 times production during these around 70 years. Fish production 0.75 to 14.16 metric tons. Egg production from 1.8 million to 114 million tons, 63 times increase. You can imagine this type of increase. And similarly, meat 1.7 metric ton to now 8.11 metric ton, around 5 times increase. So this is the status of now agricultural produce in the country and all commodity which I have seen. But just now, Professor Vasudeva has told that we are losing more than 20 to 25% agri products because of rotting and other storage problems. Therefore, the topic which has been chosen for today's discussion, nanobiotechnology for transforming food processing sectors, our actions or our future, is one of the most important topic, and it's being discussed throughout the country. And I'm sure Professor K.S. Subramanian, who is one of the experts in the country on nanobiotechnology, will definitely talk about this subject in greater detail. But to tell you what are the various opportunities and challenges for nanobiotechnology in agri-food, these are tremendous. There is a lot of opportunities which are lying in this particular field. It's a very well immersed and now important technology which we have in our hand, and it starts from crop enhancement, that is nanobiofortification, to seed priming, and then nanomaterial assisted genetic modification of crop. Then another opportunity is lying in the soil improvement, that nanoclays, nanozeolites, biodegradability or bio-nano products. And then smart monitoring. Just now, Professor Vasudeva also told that production of nanosensor for monitoring the real-time monitoring of plant physiological state of fruits and rotting materials and food products. Then fourth area, which is now dealing with improved productivity in crops through nanofertilizers, nanopesticides, and nanofungicides. When these are applied in nano form, they are becoming very, very effective. Then fifth area which has greater opportunity is targeted controlled delivery of protein-bound nanoparticles, then nanoparticles mediated targeted nucleic acid delivery for developing interspecific hybridization in crops. Then the sixth area is crop protection and stress tolerance. For pest management, we have nanobioremediation and then enhanced plant resistance. But there are certain challenges which we always foresee with all these new technologies, and nanotechnology is not different from that. And we know that the major challenge lies with the phytotoxicity or cytotoxicity of these nanomaterial, for which we need to have more studies. And then lysosomal damage within the cell, how these nanomaterials are behaving once these enter in human cells through our eatable products. And then reactive oxygen accumulation because of the introduction of these nanomaterial in our living cell system, how these are producing reactive oxygen species, which are in turns affecting our cell growth and development. Then compromised crop quality also is one of the biggest challenge which can be tackled with nanobiotechnology. Then we have very limited information about lack of toxicological evidences and risk assessment. The information in this area is required, and major centers involved in nanobiotechnology research, they should concentrate more on this area. That lack of policies, regulations, and public acceptance and awareness. This is another important area where we in country like India, the guidelines for use of nanomaterials are not yet there, but still it is being used very extensively. And Dr. Subramanian is involved in developing these guidelines, and I'm sure these would be released as very soon by the government of India. So this is a very important area in which we are dealing today, and we have seen that in spite of the fact, if we see many people, many scientists are working in this particular area in the country, but most of the projects and experiments which we have seen in production of nanomaterial, which is a repetitive type of work, we need to develop some innovative ideas, particularly for developing those nanomaterials which are biodegradable in nature. So naturally, when we are developing nanomaterials from biomaterials like agri-food waste and also agri-food residues, that material would be very important, which will subsequently degrade after its use and application. The use of nanomaterial is increasing in diverse fields, in agriculture and also in food packaging. And in food packaging, it is suggested that nanobiotechnology offers a variety of options. And one of the important options was told by just now Professor Vasudevappa, that the packing material, the packing paper, if you dip it in nanomaterial, it doesn't tear off. Therefore, this type of material is very, very important to develop, so that, and particularly the bio-based packing material is required for smart packaging of biodegradable products or material which is rotting, particularly the fruits and vegetables which rots during storage is very, very important. And nanotechnology is also used for in processed food or for their packaging, and it allows remarkable packaging material which can further protect this particular packaged material from rotting and various spoilage from the various organisms. To create new food packaging functions, the use of nanobiotechnology enables possibly improving the properties of food, such as healthier, tastier, as well as improved nutritious food when it is packaged. That is very, very important. And we know that the nanosensors, which are very, very important and intelligently used in packaging material, ultimately tells us what is happening inside the packaging material. And if food material is becoming spoiled or rotting, then immediately some color changes are happening, and it tells us in the form of a sensor that the food material or stored material is not now worth using. There are many types, more than 200 nanomaterials are available if you just Google on the net, which are used for various packaging of various material, food material, and many global companies like Amacor Limited, Sonaco Product Company, BASF-SC, Tetra Level International, Honeywell International, like many companies have come forward to produce nanotechnology based packaging materials that extended shelf life and improve food safety in various food quality materials. So there are unlimited opportunities in this particular area along with certain challenges. And if there are no challenges, then probably science will not flourish. So far, the innovations in nanobiotechnology area, I think we need to tackle all these challenges. We should identify the gap in the existing knowledge and try to fill those gaps. And I would like to say that the public-private partnership is very, very important in this particular area. And also the development of human resource, the trained human resource in nanobiotechnology is very limited. As I told you that it is only limited to development of nanomaterial from a number of starting materials or raw materials, but the skilled manpower in which the institute like NAVI, the TNAU, and also Niftem can play a very important role, so that in future we should have expert who can deal this type of technologies and will definitely use for in food processing and also food packaging area. So these are some of my remarks as an introductory or inaugural remarks for this particular area. And I would like to wish all the participants greater success in organizing this particular symposium and have very good discussion. And I'm sure that some important recommendations will appear from this particular discussion, which would be useful at national level for designing different policies. So thank you very much. Thank you, Dr. Vasudevappa and Dr. Sunil Parikh for inviting me here. And I would like to thank one and all present in this particular symposium. Thank you and namaskar.
Thank you, sir. If you have time, you can stay here for seeing these presentations. Really, your words are wisdom for us, and it will be to include in the proceedings also. And before finalizing the proceedings, I will send all this to you, and after that we can finalize and send it to the government also, so that how we can accommodate all these recommendations in our policy matter. We have first speaker with us, Professor Siddharth Tiwari, who is the scientist E at the National Agri-Food Biotechnology Institute, Mohali, India. He is representing his director also and his institute also. And as I know that he is the plant molecular biologist and genetic engineering, he contributed lot in the field, and his major contributions are towards the enhancement of pro-vitamin A content in the banana. So multiple gene constructs from Queensland University of Technology, Australia, those which are capable of enhancing PVA, were transferred in the cultivated varieties of Indian banana. In contaminated containment trials for the transgenic banana with high PVA content is being performed by following biosafety guidelines here in India. And promising banana lines will be further evaluated for bioavailability, nutritional and multi-location agronomical trials with other national institutes. And he is also working in the area of the gene editing for target banana genes, which has been initiated for enhancing the synthesis or the stability of the carotenoids in the banana. His other important contribution is the transgenic wheat lines with low phytic acid were generated by the RNAi mediated silencing of putative gene belonging to the subfamily of ABCC transporters. And the list is so long. So I invite him for presenting the work, particularly on the provitamin A, banana, and use of the genetic engineering for improving the nutritional aspects and the shelf life extension of the products, which will certainly transform the food processing sector further. So I invite Dr. Siddharth Tiwari for delivering his talk.
Thank you. Thank you so much, Dr. Sunil Parikh. I am audible.
Yeah, you are audible.
Oh, thank you so much. Thank you so much, Dr. Sunil Parikh, for introduction about NABI and about my work, whatever we are doing at NABI. So first, we are really sorry that our executive director is not able to present the work going out at NABI because he has some prior assignments in the meeting or at DVT. So he told me that I have going to be present that some glimpse of work that is going out at NABI, where that we will try to cover that all agri-food biotechnological research and innovations that we are doing in our institute. So first, I would like to give my sincere thanks to the Honorable Vice Chancellor, Professor Shri Vasudevappa, for giving an opportunity us to present our work in this big platform where we are going to introduce whatever we are doing at NABI. So I am also thankful to Dr. T.R. Sharma, who has just given us a brief introduction about the scenario about all biotechnological and nanobiotechnological approaches going on at India. And also thankful to Dr. Sunil Parikh for this presentation. So I am going to present a little bit about agri-food biotechnological research and innovation that is involved in transforming food processing sectors in India. So before going to details about my presentation, I would like to just introduce about my organization. I am working at yeah, that slides how it will move. Slide is not moving. Hello. Hello, Dr. Sunil.
We can see only the first slide.
Yeah, that this is not moving actually. Can I just again try to test this?
Yeah, unshare and after that again share.
Yeah. Just again.
Just unshare your slides from the below. Is the share option.
Yeah. But again, that it is not moving in the next slides. I don't know what is happening.
Just you can move from there only.
I'm not able to move that. I don't know what is happening. Are you able to move that slides? I'm not able to do that.
No, actually, if you send me through the email, then I can share from here.
Just I will do that. If you have any Gmail account, then I will share.
Yeah, I'm
Please send me
typing
that.
this in the chat box.
Yeah, please. Don't know what has happened.
Just try again from your side. I think now it will move.
Just a minute, I can do this. Not moving, sir.
Then send me attachment. I will share.
Yeah, please. Just have you sent mail to me? Just a minute, I will see that your mail ID.
The mail ID is in chat box.
Yeah, I have that. I have
SLIEARW.
your mail ID yahoo.co and
Yeah.
just a minute, I am doing this. Yeah, please. Wait. It's actually a big file, nearly 70. Please check that your account. I just forwarded. Now it is moving, I think so. There is some symbols like that. Now you are able to see that?
Yes.
Yeah, please.
Fine.
I'm sorry, actually in the left side there is some kind of column. It is showing that moving slides. So now I'm going to sorry for this really disturbance. This is about my institutes, and you can see that Navi is basically working, started in 2010, where we had got interim facility in phase at Mohali, and we are working on the 3R principles. That is, first is the research and development, second is resource generation, third is these two community. And following that, our vision is to provide food and nutrition security to all through agri-food, biotechnology research and innovations. So that you can see that we had shifted in 2017 in our main campus that is located in Knowledge City, where that other institutes like Business School, ISA, then Nanotechnology institutes are located. So you can see that August 2017, when Dr. T.R. Sharma was joined as the executive director of Navi, and he, along with that Honorable Minister Science and Technology Harshvardhan, and that our another director of CIAB, Dr. Sangwan, was there, and they have inaugurated this main campus in Knowledge City, where that research and administrative buildings are located. So this is another pictures of my institutes. This is, you can see that Navi is the first agri-food biotechnology institute established in India, and we have nearly 50 acres of land. There is nearly 10 acres land are utilizing for the growing of germplasm and breeding experiments. And here you can see in the pictures we have greenhouses, net houses, as well as that banana germplasm that we are growing in Navi. And we have the fields where rice and beets are routinely utilizing for performing of different breeding experiments. So Navi is working on following the mandates in the interface of agriculture, food and nutrition biotechnology. And for that, we have different institutional programs, and under each institutional program, scientists are working on their own projects. Like Agri-Biotechnology, we have project institutional program like development of designer crops with high nutrition, increased shelf life and processing quality. Then second program is genomics and computational biology for marker and gene discovery. And third one is about basic biology for crop improvement. So food and nutrition, similarly, we have another wing like food and nutrition biotechnology, where that we have programs like functional food and nutraceuticals for better health, food and GM biosafety, and another is nutraceuticals for health and human welfare. And fourth one is about that increasing shelf life and processing qualities of different fruit and vegetable crops. So just before going to details about what Navi is doing, that we have to know about the global status, where that population is increasing day by day, and that India is the just second populated country in the world, just after China, and we are contributing nearly 18% of the world populations. So it is also expected that in 2025 we will be the highest populated country in the world. So I have noted down just today's news there. You can see that fact about global hungers as well as Indian hunger, and India is just counting total 116 countries. For that, India is the rank of 101. Just you can see, and we are just behind to Pakistan, Myanmar, Nepal, and Bangladesh. Those are neighboring countries to us. And it is estimated that nearly more than 900 million people, nearly 1 billion people are hungry or undernourished. And it is also noted that highest undernourished pupils are located in India. And it was noted that out of 4 people in the world, 1 pupil belongs to India who is not getting the sufficient food in their diets. So this is the scenario. And expected that scenario regarding that global food security by 2025, that food requirements will be double, because that global population reached to more than 8 billion, and undernourished people will be more than 1 billion. So it can increase the pressure to protect the environment, biodiversity, and conserve natural resources. So how we can overcome this? There is basically 5 pillars to achieve food and nutrition security, like genetic improvement of food crops for nutrition and processing quality. Second one, where we can reduce the post-harvest losses. Third one is development of health foods and nutraceuticals. Fifth one is enabling food safety and regulatory systems. And fifth one is growth of food processing industry. So Navi is mostly working on one and all aspects, but we are going to be details about the genetic improvement of food crop for nutrition, food security, food quality, and second one, reduced post-harvest losses. So that you can again see that food processing scenario in India. India is the ranked second in the fruit and vegetable production in the world, just after China. And it was recent report from National Horticulture Board. It is noted that nearly 99 million metric tons of fruits are produced by India, and 192 million metric tons of vegetables produced in the same years. So it has also been noted down that India wastes more fruits and vegetables than whole UK consumes. So it is another report that India processes less than 2% of horticulture produce as compared to Brazil and Philippines. So that you can see that Indian scenario about food processing industries is not that much good as we should have. So how we can overcome these limitations? That we know that breeding is one of the important kind of you can say experimental things, where we can improve the nutrition or disease resistance variety of different crops, but it has certain limitations, like that variety should be close relatives, so that genes or that breeding can be possible. So we can overcome those limitations by using advanced biotechnological approaches. Again, something is there. What happened? I'm not able to move then again. I'm not able to move. Again, there are some issues. Dr. Sunil?
Yes, sir, we can hear you.
Yeah, actually, again, there are some problems because that symbol is not working. In the left side, there is symbol. It is showing that slide move, but it is again stopped. I don't know what happened.
But I have not received your presentation also.
I have sent in your Yahoo mail. Don't know what is happening because it was working, but suddenly it has stopped. Can somebody in your technical team can help in this?
Neetu, can you help?
Sir, I message you if possible, just exit with your full screen and then try. Because we don't have, we could support you if we had a copy of your presentation.
Actually, yesterday only I
So
got information I have to present this.
no
Otherwise,
issue, sir. Just try exiting your full screen, uh, out of your full screen mode, and then try moving if it works. So it has come to the title page.
Yes, sir.
So I believe it must have changed. So just try it now once more.
yeah, just a minute.
Yes, sir, it's moving now.
Yeah. Again, I'm sorry for this disturbance. Now that you can see that now I'm going to present little bit about technology developed by Nabi. I am going to present not all, but three major technologies. Those have all reached at the commercial stage. One is the colored wheat, second is edible coatings, and third one is the high resistance starch wheat. So first one is about that colored wheat, where that our colleague Dr. Monica Garg is working tremendously on this development of colored wheat that is basically rich with anthocyanin. And as we know that anthocyanins is very strong antioxidants, it can help to reduce the oxidative stress because as we know, oxidative stress is responsible for inflammation, cardiac diseases, diabetes, cancers, obesity, and aging. So what she did is she brought some kind of that colored wheat from Japan, and she has crossed those colored wheat with Indian cultivars. Those are basically very less or without anthocyanin contents. That is basically white. And after several round of back crossing or breeding approach, she developed that blue, purple, and black colored Indian wheats. And after development, she had performed some mice experiments where she has proved that this colored wheat has high antioxidant activity as compared to the white wheat. White wheat has been shown 4 to 7 ppm anthocyanin content, while the color wheat has been shown 40 to 150 ppm. So after that, she has developed different products that have been shown that anti-inflammatory effect on colored wheat by using cell lines. And also she has performed experiments in a high-fat diet mouse where that color wheat has been shown that reducing the obesity. So by proving these kind of concept by using colored wheat, she has signed a different MOU with several industries for the commercialization of this technology. The second one is about the technology where that edible coatings has been developed that is very useful for health promoting factors. That is basically arabinoxylans and arabinoxylan oligosaccharides. What our colleague scientist Dr. Kaushik Majumdar did, he has used that wheat straw that is a waste material, and he has isolated that dietary fibers like arabinoxylans and functional oligosaccharides, and he has made these combinations to utilize for food coatings. And experiments has been performed in different fruits like quinoa, apple, and peach by using this novel edible coating formulations. And he noted down that the shelf life of apples has been increased 30 to 40 days by reducing weight loss, softening, and maintenance sensory qualities. So he has also estimated the commercial value of this edible coating. What he find out that, as we know, that shellacking is the commercial coatings that is derived from animal sources, and the cost he estimated for 1% is 0.3 to 0.5 paise per fruits. While that coating has been developed by Nabi, he has estimated the cost is 0.5 to 0.7 paise per food. So it is, you can say, comparable to that commercial shellac coating that is basically derived from animal sources, while that coating has been developed by Nabi is derived from the waste material of that wheat and straw. So he has also shown that expression of interest, and several companies are interested to take this technology, especially for grapes and quinoa and apples. That technology also in the advanced stage. The third technology is related to development of high amylose wheat lines. There are my colleagues, Dr. Joy K. Roy. He has performed some mutation breeding experiments where he has used EMS, and he has generated so many lines of wheat, and he has then find out that the amylose content has been increased in some of the lines, that is more than 3 to 75% as compared to that commercially available wheat line, that is a 306. And he has also shown that the glycemic index has been reduced in this high amylose containing wheat lines. And for that he has performed experiments in mice, and as we know that low glycemic index is very important, that can slow down digested process and absorption, and it can also enhance, so that it can enhance that reduce the blood sugar content. So for that this technology is very promising, and recently Navi has put that expression of interest on 5th October 2021 for taking interest by different companies. So now I will come about the biotechnological approaches as following the today's talk that has been assigned to Professor Parikh, where that agri-food biotechnology for transforming food processing sectors. So Navi is mostly focusing on that aspects of nutrition, and for enhancement of nutrition or biofortification, we are utilizing two major approaches. One is the gain of function, that is known as an overexpression, and second one is the loss of function, where that we know that genome editing or RNAi can work. So for this we are working on here I am going to present two major achievements by Nabi. That one is the development of lysine rich rice that has been developed by the Dr. Ashwani Parikh's group, and second one is about my work, golden banana, that has been developed by us. So lysine rich rice, what he did, first he, as we know that lysine is very important, that is play a major role in calcium absorption, building muscles, protein, recovery from surgery and spot injuries, and production of hormones, enzyme, and antibodies. As we know that all cereals are not able to have that essential amino acids, especially lysine, as we know that pulses are deficient with methionine and cysteine, while rice is deficient in lysine and tryptophan. So it was recommended that we have to take the mix of rice and pulses to get our balanced diet. But our director has been designed his strategy by using biotechnological approaches, where he wants to silence some of the genes those are responsible for the degradation of lysine in the synthesis pathway, and there are some targets that he has been identified for the overexpression in the rice, like DSDPS he has identified for the overexpression, so that it has increased the content of lysine, while there are some dehydrogenase like LKR and SDHR responsible for the degradation of lysine. So he has targeted those enzymatic steps by using RNAi approach. So for that, he has basically designed 7 gene constructs, some of them by using the constitutive promoter, where CMV35 has been used, and other is where he has used endosperm specific promoter, where the genes can be expressed only in the endosperm of rice. And he has also used that overexpression strategy and RNAi based silencing strategy. So by following this, he has also designed one construct where he has used both, like overexpression, where DHDPS has been overexpressed by using that seed specific promoter, That is basically endosperm specific promoters, and also the same construct there he has used independently RNAi can also combine to develop that double construct. And after that, he has observed that the lines are very promising and have been shown that complete agronomically normal phenotypes and also the high content of lysine in that rice. And he is also able to perform that experiments where he has shown that enhanced content of lysine can increase the salinity stress tolerance in the rice. So for this, he has reported that in the usually rice seeds contain only 4% of lysine, while it is recommended by WHO should be a 5.5%. So that rice has been increased content nearly 58%, and it has mentioned that it is recommended to get a sufficient amount of lysine by using this transgenic rice. The same work he has recently published in Food and Energy Security Journals and also filed patent in 2013. Now I will come about my second work, that is basically a work about that enhancement of provitamin A or beta carotene content in banana. As we know that vitamin A is a fat soluble vitamin, and we can get this vitamin from two major sources. One is animal and another is the plant sources. In animal sources, you can directly get in the form of retinol or in vitamin A. Those are non-vegetarian, they can get this, while that those are vegetarian can get plant origin sources, that is in the form of pigmented carotenoids, and those carotenoids basically alpha carotenoid, beta carotene and gamma carotene. And as we know that beta carotene is the most active carotenoid that is going to be converted into a vitamin A in our body. And why we are working on plant source? Because it has been known that it's 375 million people worldwide, and around 30% of the Indian population is vegetarian. So we cannot ignore the vegetarian source that can provide the sufficient amount of vitamin A. So in case of India, you can see that there is a severe deficiency has been reported regarding that vitamin A deficiency, especially for children, those have less than 5 years age. And it is reported that 57% of children are deficient for this. And it is also reported that more than 3 lakh children die per annum due to the vitamin A deficiency. Especially in the world map, you can see that most of the Southeast Asian countries are deficient with the vitamin A. And vitamin A deficiency is responsible for mortality, blindness, night blindness, impaired immune system, impaired brain development. So that we started our work on banana. Why? Because banana is a tropical food crops and cultivated in more than 130 countries. Banana is the second largest produced food just after citrus and contributes 16% of the total world food production. As we know, India is the largest producer of banana that contributes nearly 26% of the world banana production. The fruit is available throughout the year at the reasonable price and most suitable and choicest foods for the diets of all age groups like infants, adults, and old peoples. And why we have to use biotechnological approaches? Because banana is a basically triploid. That means the crop is sterile and breeding is not possible. So we have to depend only by using biotechnological approach to enhance the genetic level of banana. So how much vitamin A we have to be increased? For this, we have calculated that recommended dietary allowance for vitamin A is 600 microgram. So per day we have to get 600 microgram vitamin A or retinol. The conversion ratio of beta carotene to vitamin A is reported 1:8. So if we get beta carotene 8 molecules, it can convert into the 1 molecules of retinol. So the conversion ratio is 1:8. So that if we get 100% RDA for vitamin A, we have to get 4,800 microgram beta carotene per day from the plant source. In case of banana, we had performed some germplasm screening, and that work was published in 2017, where we find out that one of the most common cultivated varieties, Grand 9, contain 400 to 500 microgram per 100 gram of beta carotene. You can see in this picture that banana figures that banana, that this is a edible portion of that is known as a pulp, contain nearly 100 or 200 gram of weight. So in case of Grand 9, as we know that it contains nearly 400 to 500 microgram beta carotene, if we want to get 100% RDA, that is a 4,800 microgram, we have to increase nearly 11-fold content of beta carotene. So by putting our this objective, by following ICMR guidelines, we have fixed our objective to at least achieve 50% RDA from single banana only. so that rest of the 50% we can get from other nutritional sources. By fixing our objective, we have started work on metabolism engineering in the carotenoids biosynthesis pathway. Here you can see that MEP pathway provides a substrate flux in the carotenoids pathway, where one of the rate limiting enzymatic steps regulated by DXS gene. And after in carotenoids synthesis, where generalized 5-diphosphate synthesized, it is going to be converted into the lycopene by the different enzymatic stages regulated by phytin synthase, PDS, JDS, etc. So lycopene is going to be converted into the synthesis of alpha carotene or lutein by the activity of lycopene epsilon cyclase, and lycopene beta cyclase is going to be convert lycopene into the beta carotene. So beta carotene our target molecules. What we did, we have identified lycopene epsilon cyclase for genome editing so that all resources can go to synthesize beta carotene, and other enzymatic steps regulated by BCH and CCDS going to be degrade beta carotene. So we also targeted these enzymatic steps by CRISPR-Cas approach. While DXS gene and PSY gene has been used for the overexpression study, and PDS we have used as a proof of concept that I will let you know in my next few slides. So these are the basic steps that we had followed. First, we had identified gene by using in silico analysis, and then identified gene has been characterized in bacterial complementation system as well as gene expression study. Then we had optimized the efficient protocol of embryogenic cell suspension culture. This suspension of embryos utilized for the genetic transformation. And finally, we had generated that lines of banana, and those lines acclimatized in the greenhouse by following the DBT biosafety guidelines. Finally, all that nearly 500 events that we had generated by using different construct has been transferred in the net houses. And after that fruiting stage, we performed that fruit analysis by using HPLC. So that PDS and LCY, as I told that we had identified target for genome editing approach. PDS we had used as the proof of concept that our genome editing is working or not. For that, because PDS is responsible for chlorophyll biosynthesis also, and it is going to be finally synthesize beta carotene or carotenoids. So if we edit or silence this PDS gene, that chlorophyll synthesis will be blocked, and also that carotenoids will not be synthesized. So we get some kind of abnormalities in plants like albino phenotypes. On the basis of that, we can say that our approach is workable to implement in the banana. For that, we had identified and designed guide RNA and CRISPR constructs for silencing of PDS in banana. And after that, we had performed a transformation by using that CRISPR construct, and you can see that albino phenotypes has been found out even during the tissue culture stages. And finally, acclimatized plants have shown that some of plants have shown complete albino phenotypes, while some of the plants have shown that variegated phenotypes. It shows that our editing of PDS is working in banana. And for the confirmation, we had performed sequencing. And after sequencing of target region, we find out some of lines have been generated stop codons in the gene by after that editing or mutation creation in that particular lines. So this stop codons are just you can say create the problems for the complete protein synthesis of frame shift mutation of this can degrade the synthesis of proteins, and because of that we find out that the synthesis of chlorophyll has been significantly decreased as compared to control. And similarly, the mutate lines have been shown the decreased concentration of carotenoids. So the same work we had published first time the Journal of Functional Integrative Genomics in 2018 as a proof of concept that CRISPR can be workable in banana. By following this, we had designed the strategy to edit that lycopene epsilon cyclase so that resources can go to synthesize beta carotene content. And for that, we had again implemented the tissue culture and transformation approaches, and finally plants acclimatized in the big net houses. And at the fruiting stage, we had when plants reached, we had also performed that sequencing analysis, find out that there is a mutations has been created. Some of the lines where that GC deletion, AGC deletion, AG deletion, and T insertion has been noted down. We had performed another experiment that is known as a T7 endonuclease to confirm that editing is homozygous or heterozygous in these lines. So after that, you can see that we had performed HPLC, and we find out that one of the promising lines GN9 has shown the nearly 6% high beta carotene content, and the color has also been changed. That is looking like that yellow colors as compared to the control. So that we had also performed some agronomical performance study in our net houses, and we find out that except one lines, those are homozygous edited lines. all the lines have shown comparable phenotypes with the control. And this is the first report in banana to improve any nutritional trait by using genome editing approach. And work was published in Metabolic Engineering in the last year. So another work is basically overexpression, that we identified this target, that is DXS, dioxazylose 5-phosphate synthase. This DXS provides the substrate flux in the carotenoids biosynthesis pathway, and we identified two contrasting cultivars, Asthali and Nenden, and the gene that we had cloned from Nenden has shown the promising results for the carotenoids biosynthesis in bacterial system. On the basis of that, DXS2 gene has been cloned in one of the vector that is known as PVI121 with the using of ubiquitin promoter, and this construct we have used for the genetic transformation in banana. And 2018 we had generated first time our lines reached at the fruiting stage, and the fruit we had analyzed, and we can notice that the content has been increased nearly 38-fold as compared to control, and it looks like completely the golden banana that has been enriched with the beta carotene content. So that you can see that when we reached up to the ratoon stage or second or third generation of that lines, we find out the content has been increased more than 50-fold, and it is looking completely golden in color. So the other cultivars Rasthali we have also considered for the enhancement of beta carotene, and the technology we had demonstrated in two cultivars has been filed for Indian patents in 2019. So then again we had used some cell line based biology study where we have used Keiko cell lines, and the protocol was optimized, and we find out that the single banana is sufficient to provide 100% RDA, because that is in the cell lines based assay we had performed by using Keiko cell lines. So the project was funded by BIRAC initially in 2012, and it is completely going to be extended in the timely progress review meetings. And now the project has been extended for next 3 years from 2021 to 2024 for the event selection trials at 5 different locations. Those are basically Navsari Agriculture University, Gujarat, then Assam Agriculture University, Guwahati, then NRC Banana, then TNAU, as well as in Nabi. So 5 locations we are going to perform event selection trials, and the purpose is to check the stability of desirable traits at different locations, then collection of agronomic yield and phenotypic data, carotenoids and composition analysis of food characteristics, and cell line or animal based biology study with the selected lines. So that our genome editing work was highlighted at different platform when it was published in 2018, and it was the first report in the world to demonstrate CRISPR approach in banana. And in the same year, it was also considered the most promising 15 technology developed by Indian scientists. In this year only that our former Minister of Science and Technology, Dr. Harshvardhan, visited to us, and he appreciated our work, and he is showing interest and supporting us to go at the next step for commercialization of this technology. Very recently, we interviewed in July for this technology in Life and Science with Pallav Bagla program. So you can say that Nabi is working tirelessly by using different biotechnological approaches to enhance the content of nutrition as the mandate of institutes. So with this, I would like to say my thanks to the organizer, and I am really very sorry that we find out several kind of disturbance during my presentation. Thank you so much, sir.
Thank you, Dr. Tiwari, for this nice presentation and given the overview of your institute as well as your work also. And this is the pioneer work done in our country. You must be send us some small write-up also for include in the proceedings of this webinar.
Sure.
Now I request to our next speaker, Professor K.S. Subramanian. And K.S. Subramanian is the pioneer name in the field of the agri-nanotechnology in India. He is the director of the research at Tamil Nadu Agricultural University, Coimbatore, India. He did his PhD at the University of Ottawa, Canada, utilizing one of the most prestigious Canadian Commonwealth scholarships. He has more than 32 years of research and teaching experiences in TNAU and expertise in the field of nanotechnology in agriculture and soil biology. Dr. K.S. Subramanian is the founder head of the Department of Nanoscience and Technology and assisted Indian Council of Agricultural Research in developing a research framework of nanotechnology platform in agriculture. He is the principal investigator of several international and national projects worth over rupees 30 crores, that is 5 million USD, funded by Global Affairs Canada, International Development Research Center IDRC, DST Nano Mission, besides ICAR. He has significantly contributed for the development of nanotechnologies for food preservation, design and fabrication of nano agri inputs, nano based on site detection kits. He is one of the core committee members in regulatory frameworks for the usage of agri nano products in the country and members in the several governing professionals and academic organizations. And presently he is serving in many of the committees for formulating the policy in the agri nanotechnology. He has published over 220 research papers in the peer-reviewed journals and 6 books on nanotechnology, and participated as a lead speaker in various national and international conferences, seminars, and he was awarded with the 14 very prestigious awards throughout the world. So with this introduction, I feel honored and proud to invite Professor Subramanian to present his work and also on the overview on the agri nanotechnology sector, which can boost the food processing sector also. So this is the synergy between the agri processing and nanotechnology also. I invite and welcome Dr. Subramanian. Sir, please share your, we cannot see your slides.
Am I audible to you?
Yeah, you are audible, but we cannot see your slides.
I shared my slide already, and I could see my slide on screen. And
but we are not able to see your slides. It is just the whiteboard only.
Okay, I'll just share my slide again. Or I have sent the slides already to you. If you wish, you can also share from your end.
I have not received the mail yet. I'm waiting for that.
I think the previous speaker has to unshare.
Yeah.
Yes,
Yeah,
here.
Tiwari, please unshare first.
Dr. Tiwari, please unshare your slide.
Yes, sir.
It's still on. It's still on the screen.
Can someone unshare my slide from your side, sir?
You can unshare. Yeah, now we are able to see Professor Subramanian's slides. Yes, sir, please continue. Now we can see.
So please unmute yourself.
What is happening? Am I audible and my slides are not seen?
Yes, sir, you're audible. We can see your slides. Thank you, sir. Please carry
Okay,
on.
but I could not see my slide. But I can keep speaking still the things are said. And it's a good event. I'm very happy that the Global or World Food Day, as part of World Food Day celebration, this seminar is organized. And I'm very happy to speak at this forum as both the Vice Chancellor and the DDG crop science set the stage. And we are in the, we are overwhelmed with the food grain production of the country, vegetable and perishable production of the country. Unfortunately, it is still, we are very low. Recently, they released statistics on global hunger index, and we are much higher, 101. It is last year it was 94, and certainly it is a serious matter of concern. We need to look at that, and today in this particular forum, I speak on how exactly nanotechnology will help in changing the agriculture, transformation of agriculture, food processing, and to show that, are you seeing the first slide?
Yes, sir.
Hello?
Yes, sir.
Yes. Okay. And this is a Tamil Nadu Agriculture University. It was built in 1909. Exactly 50 years later, it is 1959. December 29 is the year nanotechnologists always say it is a very important event. At that time, Dr. Richard Feynman mentioned about nanotechnology, plenty of space at the bottom. That was a key, the words very important for nanotechnology. Exactly, again, 50 years later, we came to existence in nanotechnology. And I feel proud that as a founder head, I'm really excited to work in nanotechnology. And I could show you how the agriculture food sector has transformed over the years because of the nanotechnology interventions. So the solution to unresolved, there are lots of global issues have been flagged off by DDG crop science, and we have answers to those. And those who are not aware of nanotechnology, I always show this particular slide for a better understanding of what is nanotechnology, why nanotechnology is very important in agriculture. And you see the map of India. Currently, the Indian population is about 1.38 billion. And if you queue them in 1 meter length, each Indian is the size of a nanoparticle. When the size reduced, the surface area gets tremendously improved. There is a unique feature of nanotechnology, and that is explained here. When the cube undergoes 24 times of division, it becomes a nano-sized particle, and you can spread one football stadium with a small 5-centimeter cube. That is the beauty of nanotechnology. And we can, because we are quite familiar with coronavirus, I can also explain how the size of the corona virus is about 50 to 120 nanometer, which means 50 to 120 nanoparticle can be assembled together to build one virus particle. And also one hair, across a hair, you can assemble 80,000 nanoparticles. All this explanation indicate that nano is very, very small, extremely small. The surface area is huge, and that is a potential we are exploiting in nanotechnology in agriculture. The small is very smart. Certainly we can think of. Another analogy to show how the word nano comes from is dwarf, and dwarf is
Excuse me, sir. The slides are not changing.
but I could see the slides changed.
Yeah, I mean, it's not showing up here, sir.
You're still with the first slide.
Yes, we are on the title slide only, sir.
I'll share one more time.
Yes, sir. Otherwise, you could try coming out of this full screen mode and try once more, sir.
Okay. Actually, I could see the slide moving. I'll share one more time.
Yes, sir. Please. Thank you.
Or I have sent the mail to Sunil already, and he can share from his side also. It was shared a few minutes ago. It's delivered already. Okay.
No, sir. Still the mail is not delivered actually.
Okay. And what should I do? Are you getting the second slide now?
No, sir. It's on the first slide only.
Okay. Are you seeing the slide now?
Not yet, sir. You can unshare and again try to share.
I think the previous
Yeah,
PPT
still
still
in-
is there.
yeah.
That is not unshared. That's the reason why it is. I don't know how to unshare the previous speaker's slide.
Shivaji, please unshare your screen.
Are you getting my slides?
Yes, sir.
Yeah.
Yes, sir.
Second slide now.
We are on the second slide. So if you could
Okay.
just try scrolling down once, just if the next slide is also
Yeah,
coming
it's
up.
moving. It's
Yes,
moving.
sir.
It's moving.
Yes, sir. It's
Okay,
moving.
I
Thank
think I
you.
explained enough how the nano, the size matters, and I explained with the Indian population, and the coronavirus size is about 150 to 120 nanometer, which means 150 to 120 nanoparticle put together makes one coronavirus. Small is smart, and nano can also be explained with the word nano refers to dwarfism, or the dwarf, which means you are aware of bonsai. It is typically a nano or miniature structure. This is what we refer to in nano. So the nano agriculture is defined as an atom by atom manipulation, enables a precision agriculture. Instead of working at the macro scale, we work at the nano scale, atom by atom, we manipulate so that the process and product would be precise to deliver input in agriculture. Without wastage of input, we'll be able to get a better crop with a minimal input. So we also get a solution to unresolved issues. This is what we do. Particularly for this particular seminar, my focus would be on why nano in food processing. There are 5 areas. Both Dr. Vasudevappa as well Dr. T.R. Sharma both nicely set the stage for this. And whatever they mentioned, I have answered to those queries raised by both of them. And post-harvest losses is a huge challenge, a global challenge that we have nano solutions. Value addition is number 2, intelligent packaging, lab-on-chip technologies for food quality, and the safety regulations that as a member in the regulatory committee, I can share how exactly nano food regulation work in India. So this is an overview of where the nano food system. This is a common picture you will see in the internet. And when you think of nano food, these are the areas we can think of: material science, the processing of foods, products, and the safety of foods. And as I said, my focus would be on 4 important areas in which how nanotechnology transforms Indian agriculture. The first one is the nanotechnologies addressing a global challenge of post-harvest losses. Post-harvest losses is huge in the country. Though we produce so much of fruits and vegetables to the tune of 326 million tons, and we are happy with the number. We are number 2 next to China. Whatever we produce at the farm, it is not reaching the people. That's why recently they made an assessment. The post-harvest losses is about 30 to 35%. In terms of economy, it is 240,000 crore, 33 to 35 billion dollar worth of fruits and vegetables are being wasted. And post-harvest losses is not a problem in India alone. Even in Western countries, the losses is in the range of 20 to 25%. Africa is somewhere at 80 to 85%. So the global average of post-harvest losses in the range of 25 to 50%. So what we have done, I had a major project funded by Canadian government, Global Affairs Canada. I got 12 crore to develop a comprehensive package to address a global challenge of post-harvest losses. So we joined together. There are 7 countries involved. I'm the coordinator for India, Sri Lanka, Canada, Trinidad and Tobago, Kenya, and Tanzania. There are 7 countries involved. Each countries work on their own fruits. And the post-harvest losses is a huge, and it happens at a different strata. The point at which the fruits and vegetables are produced at the farm gate still reaches a consumer. It has to go through 7 places, and it has to go through the farm produce going to the primary wholesale, secondary wholesale, retail, and consumer. It has to transfer to different places, and the loss is about 30 to 35%. And we need to address at all levels. Then only we will be able to minimize the post-harvest losses. In the project, what I got from the Canadian government, and we worked together as a team. We developed 7 technologies to address this global issue of post-harvest losses and how it is done. So as I said, it is loss at the time of harvest. It is at the farmyard, at the farm, we lose fruits, and we lose fruits during the transport. The fruits are not being transported properly. You look at these guys; they are sitting on the fruits. Sometimes the fruits are beaten while the transport happens. So that's a huge losses is happening. So as I said, there are several stages in which post-harvest losses have to be addressed. The first stage is the before harvest, that we call the pre-harvest. So we can give a spray. We developed a nano emulsion. It's made of hexanol. Hexanol is a plant-derived compound. That compound we made as a nano emulsion, and that spray can help to minimize the post-harvest losses. So just before harvest, 1 month before harvest, the spray is given, and the fruits can be preserved on trees by 3 weeks. And after the harvest of the fruits, fruits can stay three weeks longer. So altogether, 6 weeks we can safely reduce the post-harvest losses. The fruits that are kept under storage around 36 days or so in Alfonso mango, still they are fresh. So that's the beauty of this technology. Alfonso mango up to 4 weeks, and even we could see the fruit retention on trees is higher, and the reduction in post-harvest disease is less. And as a result, per tree yield is about 5 to 10 kg more yield could be harvested with the help of this. So this is a very interesting technology for a reason. Whenever there is a corona time, the lockdown, there is a labors are not moving. At that time, we can preserve the fruits in the tree itself. That's a good technology. It costs about 40 rupees per tree, and the benefits is enormous. So this is one of the technology we have extensively conducted experiment, and this is a technology released for adoption. And the many farmers in Tamil Nadu, even neighboring state, they also adopt this technology. Another technology is the dip. If you miss that particular part of the pre-harvest spray, we can go for a post-harvest dip. This is again a very good technology. The farm pack houses where the fruits and vegetables are assembled, and a particular point where there is the industry has a series of dipping treatments are done. So we propose this hexanol formulation, and dipping the fruits in hexanol formulation can help in extending the shelf life by 2 weeks. With the time we can go with the farthest transport. Even in the corona time, some farmers use this technology. They could, they could preserve or protect the fruits from damage during the transport. The cost of this technology is only 50 Indian pies. In terms of those who watch from United States, you can you can treat 100 kg with $1. It's a very cheap technology at the farm gate. So we also made a comparison with the different varieties of banana, and one of the varieties which was so exciting is the Nendran. Nendran variety is mainly for chips making, as the previous speaker was saying about carotene-rich golden banana. That is primarily for chips making, and that particular banana is highly benefited with this technology. So the shelf life extended about 28 days. Such a huge shelf life extension that is really beneficial to the farmers. So the hexanol, based on the hexanol, is as I told you, it is a plant-derived compound. Whenever there is a wound, this compound is used. And basically, when you bite a cucumber, when you bite a fruit, this hexanol is naturally produced. So that compound, we use it for developing the formulation. So again, either spraying in the field or dipping the fruits in the pack houses itself is a treasury for the farmer. Then we thought this is my intervention, and we developed a nano sticker. The nano sticker is made of nanofiber. The nanofiber, we made it with the help of an electrospinning machine. The electrospinning machine, it converts the liquid into solid. When the liquid is subjected to 25 kilovolt, high intensity kilovolt, the surface tension becomes zero, and the liquid becomes solid. It is a continuous thread. So we use the PVA polymer loaded with hexanol. It becomes a nano sticker. We made this nano sticker, and each box you could see at the bottom of the screen. The box of fruit, Alfonso 9 fruits in a box around 3 kilos. You put a sticker on the top. That sticker is good enough to protect the fruits for 2 to 3 weeks. So what happens is that this sticker has the nanofiber in it. It is loaded with hexanol. It is slowly released depending on the respiration rate of the fruit, as well the moisture that comes out of the fruit, and the fruits are preserved. A simple technology. So it is a very powerful technology. We extensively tested in many of the districts in the state of Tamil Nadu, and we found at least 2 weeks we can preserve the fruit with a small sticker. So this particular technology is a patented one, and we are going to commercialize this technology. And many of the private firms, they wanted to use this technology for large scale adoption. So we are in the process of commercializing this technology. It's a very simple technology. The cost of the nano sticker is just 1 or 2 rupees. Again, you get 50 to 60 stickers per US dollar. And we also did another intervention of in nanotechnology. We used cyclodextrin inclusion complex and entrapped this hexanol. Hexanol is a highly volatile compound. Technically, it is hexanaldehyde. It vaporizes quickly. So we used the inclusion complex, make as a pellet. Again, it is the same purpose. The pellet is enough to protect 3 kilos worth of 250 to 300 rupees worth of fruits can be saved with the help of a single pellet. Again, this is a nanotechnology intervention, very simple technology. The last technology for post-harvest losses, minimizing post-harvest losses is the vapor. It is again, it's a very, very interesting, very doable technology. We developed a liquidator thing. You might have seen mosquito liquidator. The same principle used here. Whatever I mentioned, the hexanol formulation, you put it in the liquidator, turn it on for 2 to 3 hours. It vaporizes and reaches 800 to 600 to 800 ppm. It varies with the fruits. And for 2 to 3 hours, the fruits can be exposed to the vapor and the fruits can be preserved. You could see the fruits that were exposed to hexanol vapor is free from diseases, particularly stem and rot and anthracnose. And we could see there is a drastic reduction in the disease with the help of this particular hexanol vapor. So all these 6 technologies, they are all powerful, they are doable. This technology is not unique to either banana or mango. This can be practiced for other fruits as well. And the response may be a little plus or minus, but we can adopt it. It is not a particular fruit-specific technology. The second set of my presentation is a value addition. As I told you, though we produce so much of fruits and vegetables, it is not reaching the people. That's why the hunger index is still, we are very high, and certainly nanotechnology will help to improve our or reduce the hunger index rating. So the second part of my talk is on value addition. Value addition could be the encapsulation of functional foods. And this is basically from the waste, as the DDG crop science was mentioning, and the nanotechnology should target the wasted fruits for their work. And this is typically the grapes. After eating grapes, you split the skin, which is rich in anthocyanin. We made it to emulsion, and when we spray dry with the nano spray dry, it becomes a powder, and that way we can improve the value addition. So particularly, we have done so far anthocyanin, carotenoids, oleoresin, curcumin from turmeric, and astrogenin. So these are the compounds that had been spray dried and with nano spray dried, and this product can be used readily marketed. And these products are very important. And also some of the, for example, this technology would be useful when the price is going down, particularly tomato. Tomato price is fluctuating a lot. Sometimes it is 100 rupees, 1 kilo, 100 rupees. The next day or a few days later, it becomes 10 rupees, and farmers cannot afford to harvest them. They dump it in the on the road or dump it in the garbage. So at that time, we can take the functional molecules like lycopene and spray dry them, make a powder, and that way we can add value to this particular crop. So another interesting work our scientists have done is the fortification of micronutrients. Micronutrients are very, very important, particularly for children and and women, and because the major part of zinc and iron get fixed in the alluron layer of the rice, and when the rice get polished, we lose whatever the iron or zinc get fortified in the alluron. So what nanotechnologists have done, nanotechnology in collaboration with food scientists, and we try to we developed some extruded rice product, wasted grains as well, and we loaded the encapsulated form of iron, which is green synthesis of iron and zinc, and put it together to become a fortified rice grains. Another interesting invention from one of our colleges, we call it a community science college. They developed a rice analog. Rice analog is made of millets. So the millets are very nutritionally rich. They are rich in micronutrients, rich in iron and zinc. And what we have, we converted the millets into rice with the extrusion technology. It looks like a rice, but by chemistry they are millets. Because the children, they hate to eat millets because the millets are rich in nutrients, but they don't care. Now our scientists have come up with the rice rice analog, which is a very powerful technology to promote micronutrient and create micronutrient deficiency as well the anemic condition. And India, lots of work is done in different pockets. DFRL is a Defense Food Research Laboratory where they fortified the milk with iron and vitamin A, and it has come 10 years ago. And the military can drink this type of fortified milk so that they get whatever the nutrient that is required. The Tata recently come up with a bifortified salt, iron and iodine, and salt. It doesn't differ from the regular salt, but they are fortified because of the encapsulation methodology. And nano milk has come, and this nano encapsulated form of products, they react at a particular pH. And of course, each area which I mentioned is all from different areas, different group of scientists who have worked in those areas and come up with this type of novel products. And recently, there is another team is working in India. It's a NIAN. There is a National Institute for Animal Nutrition. It is in Bengaluru. And scientist Dr. Hartwin, he worked on these functional molecules being fortified as in the form of neosomes. And here again, he used a nano fiber, which is loaded with some of the functional molecules, then bilayered vesicles. They all different approaches in which they loaded this compound functional molecule so that when you drink milk, you get the fortified form of milk, which is nutritionally rich. And this is one of the biomolecule resveratrol. This is the molecule which has antioxidant. You may be aware, the wine, the grape has this particular compound. The bioavailability of this compound is enhanced when it is encapsulated because as a sustained release is possible. And bilayered vesicle loaded with peptides. It can be biomolecule, any molecule. These are the general methodology. Any biomolecule of interest can be loaded, and we can add value to this particular product. And recently in TNAU, we extracted one of the active compound called astrogenin, which is a very good biomolecule against cancer. And this is the fruit, we call it as Mulsita in Tamil and Annona muricata. So this fruit has astrogenin in it, and we encapsulated, and this is in the we successfully encapsulated this astrogenin, and this compound has to be evaluated. Clinical trial has not been done, but it's a very good product. Again, just an example how it can be done. Some of the interesting point of India, we have so many medicinal vegetables, the fruits and all. So we can make value to this. Such a wonderful plant is moringa. Moringa, ginger, they all the plants and the greens and the spices that can be used to fortify our product. And one such product which we are excited recently, the Prime Minister of India, like Moringa fortified roti, which gives him lots of energy. You might have seen in the history, they say that Alexander the Great, he was defeated by Marushian, a little island, and they were eating moringa juice before fighting in the war. So that's the way history says. And again, so those who watch from DFRL, they can think of moringa being a very energetic crop. You can extract those compounds and put it as a fortified food so that our military will be stronger to fight against our enemies. Soup cube, recently there are many products have developed by TNAU, immunoboosters, using nanotechnology approach to do that. I visited one little country, Taiwan, sometime in 2012, and they made n number of products, and we have to learn from them. Though they, by size, they are 1/100th of India, and they have lots of nano products. This is one of the industry in which nano milk is produced, and nano milk, nano beer, nano wine, anything you call it in nano, they make it, and we all having the bottle of fresh juice made of nano technology in sometime in 2012. So those are the things we need to do. Of course, the safety is very important. I'm going to say a word about safety in a later slide. So the third area is the nano packaging. Again, it is associated with the minimizing the losses. Packaging is very important. We need biodegradable, intelligent packaging, IP tracking, product traceability, and so on, for which nanotechnology would be very helpful. And there are three areas in which generally people work, and we also work in those areas. The packaging could be biodegradable packaging, because many cities in India, they stopped using plastic bags. We need an alternative for which Nano has a solution. We have active packaging. We can put antimicrobial substances in the film or packaging material so that you can protect the fruits and vegetables. Intelligent packaging, without making an assessment of the quality of the produce, by changing the color of the film, we will know what it is. So biodegradable film, we developed from different sources. Some of the students are watching. So our students have developed, extracted the fiber from the banana, pseudostem. After the harvest of the banana fruit, you get a pseudostem, about 80 tons per hectare. We extracted the fiber, made into nanocellulose, which compatibilizer, we made a nanofilm, which is a very, very good film, a biodegradable film. It degrades within 3 to 4 weeks instead of 1,000 years for plastic film. We also made an assessment of the cling film, which is the conventional polythene film versus nanofilm. Nanofilm is safer, nanofilm is UV protectant, and it can also preserve fruits and vegetables. At this time, cost is little higher, but we are trying to find a way how the cost can be reduced. And another interesting point which DDG also mentioned is clays. Nano clays can be used for developing film, and you make a comparison of the traditional film where the gases exchange happens directly. And if you have the nano clay mixed in the mixture, and there is a tortuous path, and the air cannot directly hit the produce. As a result, the product get preserved, and we tested those products against some of the banana chips, sugarcane juice, and all. They can be preserved for 60 days. With these films are biodegradable as well. So they also made bottles, nano bottles, using the similar approach. So I was mentioning about nano packaging with the antimicrobial property, which means we can kill the post-stress pathogens, or the pathogen which is going to spoil the fruits and vegetables or products. We can kill them it is like shooting the enemies at the immigration. So what we do in the film, we can load with chitosan. Chitosan is an antimicrobial biopolymer, and it is extracted from the crustaceans, and it kills instantly. I think this has been extensively studied in the country. We need to look at the commercial level. And the other point is that optical sensing, on-the-spot sensing is possible. Product traceability is possible. RFID can go to the little further on nano labeling, which others cannot know what it is. but we know it can be tracked through the system. So these are the advanced development in nanotechnology, and there are some diagnostic thing. Nano diagnostics, our center also working. I have a couple of DST funded projects where we use nano diagnostic for the health status of the plant. On-site detection is possible. On-site moisture status, on-site nitrogen, and on-site virus detection is possible with the help of nanotechnology. And recently, NASF is about to fund a project on eNose, electronic nose technology, to make an assessment of the quality of produce. It could be seed, it could be food, or anything can be done. We use the olfactory response. And another Ramalinga Swami award winner is with us doing the research on paste and peel technology, where we detect the pesticide residue with the help of a small sticker attached to the fruit. Remove the sticker, go to SERS, that is surface enhanced Raman spectroscopy. Instantly we will know what is the quantity of pesticide present. And paste and peel technology is being experimented for the past 2 years. I hope in another 6 months or so we'll get the product ready to go for testing at the large scale. And this is the last one I want to say it. And nano diagnostic eNose technology. This one we did it for mangoes. Mangoes are treated with some of the spurious material like calcium carbide for faster ripening, and they do this type of mixing with the calcium carbide overnight. Next day, the consumer would not know what is how the fruit was ripened, and to do that, we used eNose technology. So we made a comparison of volatile profile that comes out of the mango fruit that are treated and untreated or normally ripe, and eNose could be done. Again, it is in the development stage and a lot to be done before reaching the consumer. And the last part is a very, very important part, is the safety of nanomaterial. And I'm very happy that the DBT constituted committee, I'm one of the core committee members, helped in developing a guideline. I think India is the first country who have a regulatory framework for the use of nano products in the country. And we started using this as well. You may be aware, the nano urea has been launched, and it has come into effect in 24th of February 2021, and it was launched, and it is across the country. People started using nano urea, and this is the safety guideline we used it. And even for the products, food products, it has to undergo the similar strategy, and we need to work on some of the microbes, against beneficial organisms. We have to, particularly when the nano emulsion is sprayed in mango orchard, we should consider some of the predators, parasites, particularly honeybees. Honeybees are a very good indicator of nanotoxicity. And these toxicity guidelines were developed based on OECD protocols. And the guideline is an open document. It's a document. It's in the public domain. Anybody can refer. All these details are given. So we also look at the safety against earthworm, safety against aquatic system, and finally the human consumers. And unintentionally, sub nanoparticles get into the human system. It is likely to get accumulated in 3 places: epithelial cell, liver cells, and adipose tissues, and so on. So those are the likely places toxicity is going to occur, because those are the point in which it get accumulated. And we have to do this as Deputy Director General ICR, Dr. T.R. Sharma was mentioning about cytotoxicity and genotoxicity has to be assessed before doing that. Right now, our center is recognized as one of the centers for toxicity study as well. And other than that, there is IITR, Indian Institute of Toxicological Research in Lucknow. They also do the toxicity studies. And this is our laboratory, and all the machines are assembled under one roof. And this laboratory has a ability to do the synthesis of nanomaterial, characterization of nanomaterial, use of development of products, and value addition of products, as well as biosafety. And this facility is open to all. Anybody can use it. and on cost basis we do. And we grew up very fast, and we started with 30 lakhs as a founder head. I could see there is a 100-fold increase in terms of financial support from various sources. As a government organization, we know how funding is very important. We reached 30 crore. We started with 30 lakhs. 30 lakhs turned to 3,200 lakhs. So that is a potential of nanotechnology in food system. And some of my work has been referred in Nature as well in 2017 and 18. And these are the products TNA has. We have products for fruit preservation and nano sticker, pellets, nano film, and so on. And we also have the nano product for agriculture, the seed coating, designer seeds, and we have Rhizo Boost to improve the health of the seedlings, and we have leaf, leaf, that is for the stem borer, and we also have the diagnostics. And recently we released one sanitizer as well against COVID-19. So the best part is we have got the regulatory guidelines in place. Certainly anybody can use this particular guideline, those who wish to work in this area. These are some of the development. As I said, I serve as a core committee member. Nano fertilizer came into the picture, came into existence, and people started using it. And there is a FCO regulation, fertilizer control order, under which nano being referred as a subclass of FCO. And these are the books I have written on nanotechnology in agriculture. These are the funding agencies. I have to thank ICR because they gave me lots of support at the beginning. They also made a national convener, and that helped me to scan through the country where nanotechnology can work. We started as early as in 2010. It's about 11 years now. TNAU, NABARD, they gave me NABARD chair, Ramalinga Sametri and fellowship, DST, DBT, and CERB. IFCO is giving funding for nano delivery system. We started using nano fertilizer, using drones. There are 2 researches going on, and they supported, and major funding comes from the Canadian government. It's about 12 crore. Altogether, it's 32 crore. So I conclude my talk by saying that the traditional foods, whatever we are eating as biryani or Colombo, sambar, and all mixing together, has all transformed into the capsule type of food. Then we can nourish our children perfectly. And these are the food, they come from DFRL, Defense Food Research Laboratory, where most of the foods are very condensed, concentrated, and they help the people to grow better. And thank you very much for the opportunity given. Those who prefer to communicate with me, my contact details are in the screen. They can make a note of it. Thank you very much, organizer, the Vice Chancellor of Niftem, Vasudevappa, for giving me opportunity to share my thought in a very important event like a World Food Day. Thank you very much.
Thank you very much, sir. It's unmatchable work done in India. And we see that how you are working in the agriculture to the processing and then the fortification work, and work is again going on. So we are thankful to you for such a nice presentation and your such a nice work done in the country. Sometimes ago, Professor Gopinathan Paliat from the University of Guelph has given me your address and your phone number, and he said me that I should contact you for collaboration purpose. And definitely we will send our team to your center also for visiting, as we are now starting the nanotechnology work in the food processing center. We will talk to our vice chancellor, and after that we can plan everything there. So once again, I thank you so much, and we will take the question after the 2 presentations. So you must be stayed here. I think that there should be some questions for you there. And now I invite Dr. Asghar Ali, who is from the University of the Nottingham and professor in the post-harvest physiology and technology. Professor Asghar is the founding director of the Center of Excellence for Post-Harvest Biotechnology at the University of Nottingham, Malaysia Center, a research group is deemed for great success in its field and great impact beyond that. With a range of the post-harvest students under his guidance and supervision, Professor Asgar has set the grounds for pioneering and innovating research in the field of edible coatings and films of post-harvest biology. Additionally, he is constantly involved in the progress of science within post-harvest biology as an editor, associate editor of the Journal of Horticultural Science and Biotechnology, and member of the editorial advisory board of Asian Journal of Microbiology, Biotechnology, and Environmental Sciences. He has published extensively, particularly in the Journal of Post-Harvest Biology and Technology and other similar journals, and he is now the name synonymous with the nano edible coatings. So I invite Professor Asgar Ali for his talk on the nanotechnology, or particularly the edible coatings and nano edible coatings, which can reduce the post-harvest losses in the fresh produce. So welcome, Professor Asgar.
Thank you,
I
Professor
think
Sunil Pari, for giving me a nice introduction. Can you hear me?
yes, yes.
Can you ask him to close his slides? Dr. Subramaniam, I think his slides is still there. He's sharing the slides, so I can share mine.
He has left, but how the slides are shared still, I don't know. He is not in the meeting and he has shared. He has exit. But still we can see his slides.
Can you see my slides now?
Yeah.
Yeah, I think I have. So, all right, Mr. Sunil. First of all, I would like to express my sincere gratitude to the Niftem, to the Vice Chancellor of Niftem, Dr. Vasudevappa, and Dr. Sunil Tariq for inviting me to give a talk on this platform. So let me introduce to you that I'm Professor Asgar Ali from University of Nottingham, Malaysia. I'm going to talk about the role of nanotechnology in reducing harvest losses and quality of fresh produce. And earlier, some people have already mentioned some of the things. What are the implications of post-harvest? We know that there are going to be a 10 billion population, so how we can feed important now. And just looking at the one strategy, growing more crops in non-sustainable manners is going to be very critical. So other ways that how we can reduce these losses and enabling them to have more food to feed the current population. And we all know that there are enough food to feed the current population, but we are not getting enough food because of the lack of availability at all the places. So before I start, I would like to talk a little bit about my university. This is the University of Nottingham, based in the UK. It is based in Nottingham. It has 5 campuses abroad. 5 campuses and 3 campuses in the UK and 2 campuses overseas. So this is the first university who has a full-fledged campuses in China and Malaysia. This is just an overview of China campus, and this is the overview of Malaysia campus. So you can see there are 2 things are very common here: the clock tower, and the lake. You can find in all the campuses in Nottingham. That is the iconic thing for University of Nottingham. This campus was opened in 2000. Yes, full-fledged campus in 2000. We are not very old campus here. So you can see this lake and clock towers are iconic things for the University of Nottingham. So this is the center. It was inaugurated in 2010. With the help of Professor J.D. Roberts and Professor Graham Seymour. You can see that he has played a very instrumental role in favors of a tomato, and Professor Greg Tucker. You all know that Professor Greg Tucker works a lot on the cell wall enzymes on tomatoes. So this center actually was open in Malaysia because in Asian countries we need. Basically there is a lot of wastage in the Asian country as compared to the UK and European countries. So that's why the center was based in Malaysia with the intention to reduce the post-harvest losses and also to look at the technologies which can, feasible technologies which can reduce the losses. And also, it's also one of the other important things is that to look at the alternative management of diseases for the fresh produce. So what we do is we combine fundamental science, we discover things with the applications. So we focus on more on application side of things, where there's the problem in Malaysia, region, and the world. We combine the fundamentals and application side of things. I think in the first lecture, I think he has already mentioned this is a very tremendous work done by the crop scientist. You can see that it's a major improvement in the food security in the last half century. In 1960, you can see that the cereal yield was like 1.4 ton per hectare. But in 2014, it was increased to almost like double in 4 tons per hectare. So this was just like a double the production of these cereal crops. So food security was improved significantly. There is no doubt that this crop production was increased. So what is the worrying thing is that if you look at my right-hand side, you can see that how much global land available for these four crops. If you look at it here, 1961 to 2000, you can see that the global land area was increasing, and after that it stagnate and almost there. So we do not have much more land, arable land to grow the crops. So that's why we need to look at how we can improve the food security using different means. Other like world food supply is also shrinking because of global warming, balancing populations, and a lot of wastage, and also a deterioration of environmental concern. And also the food is running out. The food is running definitely is running out. You can see that by 2050, the population is almost like 10 billion. And this is the supply and this is demand. This graph shows for produce, which is most of the populations rely on that. So you can see that the green color is a supply and the red color is a demand. There is a big gap in supply and demand in the yield of these major crops. So, and it is more into the rice, because you can see that the rice is becoming more popular than the wheat, and the gap is also becoming bigger and bigger in supply and demand. So these are the challenges we have. And above that, we are wasting 1.3 billion tons, which is about the 1/3 of the food we produce. So I think it's like everything kilo in 10 kilo, and this is a very dramatic study. And that's why this has become one of the global challenges and leaded by the United Nations. How can we use by half by the 2030? So this is what we have to think about. That this is what we have to look at or formulate this strategy to combat this problem. So you can see that this is like a 1/3 food, and it's also come with the geography. There are several reports coming nowadays. So the problem here is the loss is not inevitable. Right? Last decades, especially in countries, people were thinking that this is inevitable, so we can't do that. But if you look at the current trend, it's not like that. In the developed countries, there are technologies, there are manpower, there are infrastructure which can reduce these post-harvest and food waste losses. The only thing is that we need to have a strong policy, that we need to have feasible technology, we need to have a good infrastructure to reduce this losses, and that is not very difficult. So basically, it's like a supply chain. This is what we are growing, and this is what we are getting as a consumer, and we need to improve this supply chain in order to have a good food security. So 30%, if you look at 30%, is like a very conservative data. If you go back to the small class countries, something like a Fiji and Southeast Asia, Indonesia, Malaysia, or India, sometimes the losses exceed 50%. What we need to think about that. So I think somebody also mentioned earlier that this is a very contrasting scenario. We say that we do not have enough food. Look at this trend. Like 50% population is overweight or obese, which is about 1.2 billion, as compared to the undernourished people, which is about only 87 million. It shows that there is not like that there is a lack of food. The only thing is the lack of availability at all the places. So this is what we need to think about that. to avoid this contrasting scenario. When we waste food, it's not only wasting food. So this is the cost came out. It is about $2.6 trillion. It includes all these, what we use for energy, what we use irrigation, pesticides, all the chemicals and the laborers. So all the costs like $2.6 trillion US dollar, which is about the economy of the many countries. Not only that, if you look at the environmental concern, we are losing 3.3 billion metric tons of carbon dioxide just coming from the food waste, coming from the food waste where we dump it. That is also a very challenging area. How can we reduce? If we reduce the food waste, what we can reduce the CO2 coming out. So if you look at this global food losses, you can see like 30%. When we say about 30% of food loss, it's not like only the food, and the value is about 1 trillion more than, and it's not only food, we're also losing the nutrition, we're also losing the land, water, chemicals, energy, and employment, and also coming from the contaminations. So if you look at this graph on my right-hand side, so this is what we are losing it, consumption level, and this is a distribution level, and the red color is the post-harvest level. So the cereals, you can see that the post-harvest level is high because of the lack of harvesting techniques and other things. The same thing goes with the roots and tubers, and you can see that the consumption level is not that much as compared to the agriculture and the post-harvest losses. Look at the fruits and vegetables, it's a big loss as consumption level as well as in the processing and the post-harvest level. So some of the examples I'm giving you here, like in the UK, which is a very small country, it's about 5 million tons of food waste annually, which is about 85 kilograms of food per year. That is enough to feed the 15 million population. You can see, and this is wastage resources, water and nutrition, chemicals and labors. How much chemicals we are dumping into the resources, dumping into the environment, that is another matter of concern. So this is something about how much we're losing in India. This is a great pride to see that we are the largest producer of food after next to China. So, and somebody also have presented earlier that we are producing like 280 million tons of grain, 300 million tons, 300 million tons, which is also, still we have a deficit of 20 to 25% per year. We follow the guidelines of WHO, about 400 grams, consumption of 400 grams of fruits and vegetables by each person per year. But out of that, in India, we are losing like 30 to 40% of fruits and vegetables, which is about 1.3 lakhs crore annually. Can you see that this is a huge amount? And these are the major 4 states like West Bengal, Gujarat, Bihar, and UP. About 13,600 crores to 10,300 crores in UP. And one of the reasons what we have found that, out of 300 millions what we are losing, only 2.2% of these are the processed. As compared to USA is 65%, China is 23%, Malaysia is 15%. Can you see that this is an area where we need to think of that? We have to increase our food processing industry. And this is not a bigger challenge because we can start at the very low level, village level, or I'm sure the ICR or other institution in India might be doing something about establishing a small scale factory where it can process the fruits and vegetables locally in the form of to add the value and then sell them in the market. So this is a scenario we need to think about, that we have to increase the processing from 2.2 to 4. And also second issue is that infrastructure such as cold storage. You know that in India, the major cold storage are used by potatoes, which is about 370 lakhs tons for fruits and vegetables. We need to build, we have to build a cold storage for 370 lakhs tons for fruits and vegetables. This is another thing we need to think of, that it can only possible by capacity building and also the robust policies. You can see some of the Indian farmers face 93,000 crore post-harvest losses. It's another one, 2 trillion. India is doing 2 trillion per year post-harvest losses just from fruits and vegetables. I'm not talking about the grains and other things. So this is, you can see there are what and how much India wastes. Like cereals is like about 5%, pulses is about 8%, oilseed is about 9%, fruits and vegetables about 16%, and milk is about 0.92, meat is 2.7, and poultry meat is 6%. And this is like, you need to think about food security versus the food wastage. And annual wastage in India itself is like 92,000 crores in 2016. If you look at the grains and also the vegetables, there are different categories. You know, we are looking about during the operations. I think we do not have much better options to deal with this fresh produce. But this is another way we need to think about that. This is the one data I found that like out of these all commodities, the major commodities and vegetables, because of highly perishable in nature, is about 20% of the conversion. One of the scenarios like lack of storage facilities in tomatoes. Okay. So now come to the point we need to focus on fruits and vegetables. Like out of the food waste is like 30 to 45% is coming from the food. If you look at this food in Europe, which is about, you know, is a very less losses as compared to Western Sahara, Sub-Sahara, and also the Asian countries. And you know, one of the reasons we didn't focus on food waste is you can see it here. Like we invest 95% of agriculture resources in the production of food, while only invest 5% of food preservation. So this is something we need to think about, that we need to increase this investment for the food processing industries. So if you look at crop preservation, we use only 5%, and crop production until now we have invested about 95%. You know, this is scenario. I think it has been changed now. Like Bill and Melinda Gates, Rockefeller Foundations, and major NGOs, they are started investing recently. Like the Rockefeller Foundation has invested $30 million in Nigeria for tomatoes, mango, or in Kenya for mango, and to other sub-Saharan Africa. So these are the investment they started doing it after looking at the scenario of this wastage. So what we need to do that, I think one of the very important thing is like we need to fill up this global post-harvest, the skill gap and technology gap. There are two things. Means that we do not train our people. We do not teach at school levels. We teach at the university level. No, but now many universities are teaching this one. But how, what they are teaching, whether they have changed their syllabus to make it in a better form, or they are still teaching the very old technology, harvest technologies. Second thing also, we need to technology gap, you know, so we need to invest money in our post-harvest technology to fill this gap. So what we do at University of like you can see that this is the group of students just looking at a challenge mapping exercise for food waste and food losses. We are educating young post-harvest students. So this is a module I developed with educate young students, and also we provide training in preservation technology for some countries, and also this is coming to us from different countries. And also conducting research and developing innovative technologies suited for the preservation of food in developing countries. In developing countries, what we need to do, we need to look at the feasible technology rather than any other technology. I think it's not going to work in the. So some of the technology I mentioned it here, which is like very traditional technologies used in the Asian countries. We need to change and make it more feasible technology. Our major problem is the lack of cold storage and different containers. So let's come to the points like tropical fruits and vegetables. These are the major play in the food supply chain. And there are two problems. They have very short shelf life and also the post-harvest diseases. The short shelf life due to the water loss and accelerated ripening. And post-harvest diseases, I think the most important disease is anthracnose. I think the previous speaker has already mentioned that about the post-harvest about the anthracnose. And this anthracnose disease is a very devastating disease. You name the crop, it will infect the tropical crops. So mango, papaya, bananas, all the tropical crops are infested by the anthracnose. Second problem is that it has a quiescent infection. People don't know about that. So we need to find out the early detection technologies to know that this disease is there. So sometimes we harvest the food, put in the cold storage, and where you can see the disease appearance there. And this can be controlled by chemical fungicide, but most of the chemical fungicides getting resistance for this disease or for this fungus. So fungus are getting resistance. There are health hazards. People have found that tetragenicity, next generations, and there are birth defects due to the highly use of these fungicides and microbial products. Another thing, the third point is like environmental concern. We are dumping too much chemicals to the groundwater. So this is anthracnose disease, like I mentioned. You know, there are several species, but two are the major one: Biotechnological Approaches, which causing papaya, bananas, or dragon fruit, and these are the anthracnose, Biotechnological Approaches, and mosaic and capsicum, which is in in fact, in bananas, bell peppers, and other crops. You can see this is a pure culture of Colatotrichum globosporidus, and this is another culture of Colatotrichum capsicum or mosaic. So this is I'm going to talk more on about the dragon fruit, and you can see the dragon fruit nowadays is a superfruit in the tropical regions. So I'm going to talk about novel approaches, which is like use of natural products and enhancement coatings. So I'm not going to talk about traditional coatings. I'm going to talk about the nano coatings. So just to inform you that what the coating is, edible coating is that is the materials used for enrolling various foods to extend the shelf life of the product that may be eaten together with the food or without further removal or considered as an edible coating. And second issue is that there's a lot of plastics wax is coming out, like environmental protection agencies says that every year we are using 1.6 million metric tons of package waste dumped into the landfills. So plastics are non-biodegradable, and today I've seen that France has already banned this plastic material for some of the fresh produce, and they are going to ban it completely by 2023. We need to look at the alternative ways how we can enhance the shelf life biodegradable materials. So this is a bit of edible coatings, like these are biodegradables, environmental friendly, they are ripening, they have antimicrobial properties, slow down the gases exchange, and this is how it deforms the semi-permeable membrane on the food surface. So you don't need the packaging materials. You can use the coatings which can modify the atmosphere, and somehow it will also protect from the abrasions and provide resistance to the external environment. So I'm going to focus more on chitosan, and previous speakers talk about some chitosan. So how we produce chitosan, we make it nano or some micron dispersants from the field level to the post-harvest level. So if you don't know the chitosan, this is the chitosan. This is second most abundant polymers found on the Earth's surface. And this is this is a waste material, totally waste materials. And this is a very versatile product, I would say that. Very, very highly versatile because of its nature. It's highly polycationic in nature, and that is how it attracts the negative particles of DNA problem, the microbes, and then it shows the antimicrobial activities. It can be applied through soil, foliar applications. It also has a fungistatic and fungicidal. Most important thing is induced resistance, so it will, besides the activities of antifungal hydrolysis and total phenols, it can fight against the plant, against the diseases. That is a very important issue, properties from this chitosan. So chitosan is not, you can see that from the scale of this seafish industry, we have a chitin, and after deacetylation, removal of acetyl group will become chitosan. You can see that is chitosan powder. So the only cost involved is the removal of the acetyl group, that is a little bit costly, but now there's a lot of new technologies which are making very, very, very reasonable. It also found the semi-permeable membrane to reduce respiration rate. So this is, you can see that this is a chitosan application. We were working on papaya and bell peppers, and it's already commercialized by some company here. And what we have found that, what we have found that when you lose the traditional coating, the problem is sometimes it happens too much on the food surface, so it can completely inhibit the respiration rate and its saline evolutions. So we thought of that making into nano form, so it will have a better effect. So you can see that this is 3 days after inoculation of this Colatotrichum globosporidus on the what is called dragon fruit plants. So we started post-harvest first, and after that we thought that we got another project. Why don't we start from the seedlings and then continue post-harvest and see their effect? So this is a dragon fruit. You can see that. I think many of you know that. I visited last time to Trichy, one of the regional stations in Tamil Nadu Agricultural University, and found that on the way to Lai Canal, there was a lot of plantation of dragon fruit, and it was really expensive, much expensive. So as you know, that this is a very superfruit. It has a very high antioxidant properties. It contains like vitamin C, beta-carotene, lycopene, and it's considered. And the other thing is it's very high. So this is a very superfood. The major problem is that is the anthracnose disease in this. So, like I said, we started with edible coating. So, what are the limitations of using conventional chitosan is that the concentration lower than 1.5 proportion lower than 5% has limited effect on the control of microorganisms. If you increase the concentration, then it blocked the respiration of the plants and resulting in the death of the plant. That was the issue with dealing with the guidance. It's very difficult to make a solution more than 2% because it's highly viscous in nature. We use high power stirrer to make this solution. You know? So, like I said, we started with the conventional coatings in papaya, and you can see that when we use that, 1.5% is a very good quality. But the problem is if we increase the concentration, you can see this is the papaya. There was no disease, but when we realized that it failed to ripen after a certain period of time because it's complete, and rather than ripening, it gone into fermentation stage. So what we have found that using conventional coatings, it is completely reduced respiration rate. So completely reduce the respiration rate, and also there was no ethylene production at all. That is why it failed to ripen. Then I have spoken to one of the scientists here, chemical technology guy, and he said if we can make it into nano emulsion. This is we make that was a nano emulsion with the aim of that and it can penetrate through the skin. So we thought that it also have, like I said, a fungi-static problems. Static, and also it has like something like it can penetrate and it can induce resistance in the food surface, and they can fight against this fungus. So it's just like what we are using for this COVID-19. We are taking like vaccination, so something like that. It can penetrate into cells and it can induce resistance to fight against this Polytrichum virus, and it can also increase surface area and resistance. One important thing that it will start coagulating it. So when we make nano emulsions, the problem is solved with them. This is how we prepare this chitosan nano emulsion using Bridge 20 and Bridge 56. This is another issue, like you need to use a good emulsion. It varies a lot, its properties varies a lot. This is the final dispersion to maintain the pH about 5.6, because when this is only dissolved in the organic acids. When you use organic acid, it drops down. We need to maintain the pH. So these are the factors. You can see that this is how it looked like Polytrichum in the plants of trans. So there was a time when we got this project, like most of the plants in Malaysia were wiped off because of this disease, and it was not controlled by Benobil and any other rates. You know, this is, you can see that this is the infection of Polytrichum. So what we screen, so like I said, like we were working on different kinds of chitosan. We have done a huge amount of work on chitosan using different types of chitosan, different source of chitosan, different deacetylation percentage of chitosan, different types of chitosan. We have found that after we use different low molecular weight, medium molecular weight, high molecular weight, and also compare the locally prepared chitosan, which is much cheaper than the used by the Sigma and other companies. You can see that we compared all these chitosan locally, high molecular, low molecular, and this was the effect, concentration of the low molecular weight and locally produced chitosan. We found that locally produced is equally good, or some extent better than the chitosan produced by Sigma and other companies. You know? So what we have chosen low molecular weight, chosen the chitosan locally prepared as compared to the other chitosan. So on the basis of these results, we chose low molecular weight chitosan and locally prepared were selected for the sub-micro. So we have different concentrations and different temperature, same temperature, different sonication times and emulsifier, and different droplet size from 600, 400, 200, 1000. And you can see different droplet size and different concentration of chitosan. We tried on this dragon fruit. You can see that like a concentration, we increase the concentration, and we also compare with different nano size of these, and you can see that this was the results. Like means increasing the concentration have not much effect as compared to decreasing the size of the this chitosan emulsion. So you can see that like all these 200, 400, 600, 1000 nanometers, and you can see the different concentration. And what we have found, we have selected this one, you know, 1% to 2% and different nano size, because we all have very similar effects. So we have chosen the lower concentration for the next of the results. So what we have found that you can see that, when you look at the mode of action, this is the control, you can see the germ elongation. And this is a very normal hypha growth in control. But in the treated conoidea, there was no elongation of germ tube. You can see that there were morphological changes. They didn't allow to grow at the in vitro studies. And this also hyphal agglomeration, when we use 1% chitosan and 600 nanometer of droplet size. So these are something like look at the mechanisms on the mycelium growth or spore viability of Pleurotrichum, and you can see that this is the control in the blue color as compared to this is the different size. So red is 200 nanometers and this is the 1000 nanometers. So you can see, and this is the spore viability. The spore viability you can see totally reduced when you use the 1%, 1.5, and 2%. And you know that the Pleurotrichum is a very versatile and very dynamic fungus. It's not easy to control. But this nanoemulsion are able to control manage in vitro. And what we found that how does it work? This Pleurotrichum basically is make the food soft or make the plant soft by producing a lot of beta-galactosidase, enzyme which is responsible for dissolution of cell walls. When we use this one and if we found it on the plant, you can see this is a control micromoles as compared to the different nanometers. So you can see this is a conventional one, and this is the control. Conventional also can reduce the beta-galactosidase activity. But if you look at the 200 and 600 and 1000, they have significantly reduced these beta-galactosidase, which was produced by the Pleurotrichum spiders. So based on that, what we have seen, we have tried different concentration, different size on the plants. We are talking about in the field, talking about disease progress curve, dragon fruit. This is after 90 days after, and this is the total area under disease progress curve. And what we found that the 600 nanometers, 1% chitosan have the lowest disease incidence and the lower total disease progress area, 5.44 hectares. And you can see these are the plants. You can see that this is a C plant. This is a picture at the C, which is about treated with the 600 nanometer droplet size at 1%. And these are 2% conventional chitosan, and this is 2% 600 nanometer of chitosan. I think if you increase the size of this, it doesn't work on the plant what we have found, because it is trying to cover the skin too much that they have some phytotoxicity. They reduce the photosynthesis process. It forms the semi-permeable membrane. It forms the membrane which is too thick. It's not allowing plants to look at the allow plants to go for the photosynthesis process. So what we found 600 nanometers at one plant and the in the plants in the field, and you can see that this is on the disease severity, which is score 1.6 and 25 as compared to the control, which is about 3.2 score in the plants in the field itself. So one of the reasons is how does chitosan work? This nanoemulsion is several ways, but two major ways control disease. One is that it is in the terms of it has the inherent properties of fungal or antimicrobial properties. And second thing is inducing resistance. And when we found out what makes them to induce resistance against this fungus, one of the things is total phenolic contents, total phenolic contents. And you can see it here. It increased the fold. You can see this is the control, and this is the 600 nano droplet size. It increased several fold increase in the total phenolics in the plant itself, with inoculated and non-inoculated in the both places. So inoculated and non-inoculated give the better results. Comes up producing more phenolic, which is antimicrobial content in the antimicrobial for this fungus. So we continue after that we harvested the fruit and keep using some micron chitosan dispersion on the post-harvest of the fruit itself. So we also tried, 3 different types: low molecular weight, and this chitosan concentration of different concentration, and is the different droplet size. And we again isolated from the post-harvest phase from post-harvest fruit. We isolated, is there any difference in this species? Is there any difference out any other variants here? So, and we look at our late cordial germinations, and I'm not going to do all the results because of lack of time here. And we did the same things what we have done it. So you can see that this is a Biotechnological mosaic, and this is on dragon fruit, and this is the treated one. So you can see there's several morphological changes found in the spores or conidia of these Biotechnological spiders. These are one of the reasons it doesn't allow them to grow at the first place. And it is, even if you look at the traditional one, it also didn't allow them to grow in vitro or in vivo, but the some micron one is basically more effective as compared to the traditional one, because I think it can penetrate. So if you don't know more about that, you can go into some paper. They totally change the morphology of the conidia. That is another mode of action. You can say that this is a controlled fruits, and this is the fruit treated with 600 nanometers at 1% phytosan. And these, if you look at the morphology of this dragon fruit, it's very different from the apple and other fruit. So if you look at other technology, which is not maybe effective, but if you put the add glucose cover, and this is the fruit which lose water very much because of the from the calyx reason. When you put the fruit, you also look at the water loss, and that is a significant extent. Okay. So if you want to read more about that, you can go to this paper and read more about how does it work on the dragon fruit. So on the basis of disease results, only 1% conventional phytosan, 200 nanometers and 600 nanometers sizes at 1% phytosan were selected for this post-harvest test. So we didn't continue with we narrow down the concentrations to work on the post-harvest one, and this is what we have done. You can see that when you, like I said, the weight loss, this is the control fruit, and this is the 1% phytosan, conventional phytosan, and this is the blue color is 1000 nanometers, and the pink color is the 200 nanometers of droplet size of 1% phytosan. So you can see the significant reduction of weight loss by using that. But highest, of course, in the post-harvest phase, this is the highest one found in the conventional phytosan. You know why? Because if you make it nano emulsion, it is basically it mostly hits. So what we have found that when nano emulsion is very good in controlling the disease, whereas the conventional one is very good in controlling the post-harvest. So because of this one, and if you look at the firmness, we measure the firmness. So still, still the conventional phytosan is the best one is controlling the food firmness. The nano emulsion also have a better effect on maintaining the firmness of the food as compared to the control one. So, and you can see this is the total phenolics, several fold increase in the total phenolics. This is the 1% conventional, still and 1000 nanometers are the highest one in preserving the total phenolics and the flavonoids, because dragon fruit is highly rich in flavonoid content. And you can say this is control, and this is the 1% conventional phytosan, and also the 600 nanometers, and this one. So treated one have a similar effect. There was not much difference into the nano size, except the very too low one, 201, pink color one. This is a lycopene content. They have a very high amount of lycopene content, and this is a very similar effect on the dragon fruit. There is very little lycopene content at the 21 days, but whereas very good in the conventional phytosan and also the 1000 nanometer of droplet size. Same thing, look at total antioxidants, and were found by using the nano emulsion and the conventional one. It is very good in preserving these all these phytochemicals in the fruit itself. I think one of the reasons is to reduce by reducing the respiration rate and delaying the whole process, or delaying this ripening process during the post-harvest life. So what findings in this project is from this, what we found, low molecular weight coefficient showed the best antifungal results as compared with the high molecular weights and all those things. And 6% nanometer droplet size and 1% phytosan concentration has been reducing disease incidence by more than 80%. Well, one of the reasons is that because of they elicited the production of total phenolics, BPO, and polyphenolic oxidases, help in the induction of the lignification of this plant as well as in the plant, and that can be maintained after the harvest in the food skin. And what we have also found that the submicron phytosan this question also helps in enhancing the vegetative growth because of the maybe content that. So they were more healthy, they were more photosynthesis in the plant treated with the some micron phytosanitary. But it's only the lower one, 200 nanometer, but not in the one with the traditional chitosan. So, however, the conventional chitosan in 1% has the retention of the food quality during storage much better than the nano emulsion, but the nano emulsion has a better effectivity in the controlling the disease. So, just to show you that we have also done on tomatoes, and it also gives a very good results, like sound assisted, this kind of nano emulsion. And you can see that this is we also done on the papaya, so it also have a similar trend. The weight loss and the firmness was maintained, and weight loss was reduced when we use phytosan that we're putting on that using the phytosan coatings. So we also look at what is the thickness of this one. So we done some SCM studies, and what we have found that the thickness is about 100 micrometers. It's about 100 micrometers, and you can see that this is a treated food and this is a non-treated food. You can see the severe destruction of the food surface. Of course, you can see that this is on the food surface, and that is the place where they are losing the more water from the skin surface. And somehow it is protected with the treated one. Very smooth surface on that. There was a less transpirations were occurred during the storage. And this is the
excuse me, we are already running short of time.
Okay, I'm just going to wrap up now. And this is the we found that the skin thickness is 2.19 micrometers, 2.7 micrometer forms of the surface. So, like I said, there was one problem with when we use high concentration chitosan, there was no production of ethylene. No production of ethylene when tried in papaya. So that is why they fail to ripen. So we have to be very careful when doing this coatings one. So we also look at the internal gaseous atmosphere, and what we have found that there was no ethylene, there are presence in the food surface. They fail to ripen on that. So this is you can read more about that. We also did on papaya. There was some I wanted to show that we also make an nano-emulsion from propolis, and you can see that we tried on banana, and we make it composite like chitosan, propolis, and all these things, and we found a better result. So we, in our lab, what we are doing that we step-by-step approach to enhance the efficacy of these animal coating, enhance the efficacy of natural product. Of course, it's working, but if we can increase to next stage, we can increase to next stage. This is what we are trying to do that. We also found that when we use ethanolic extract, there was some phytotoxicity. It's not disease, but there was some phytotoxicity when you use high in concentration. So we tried to make it into the gum arabic and some essential oil. So this is all my project gum arabic and we tried on chilies. We found a better effect on nano-emulsion when combined both three products together. So that's all. This is what that we're putting prepared from natural compounds, environmentally friendly and relatively inexpensive technology to extend the shelf life. But what is more important that this is we need to make it tailor-made because of the different morphological characteristics of the fruits and vegetables. We need to make a tailor-made for the some of the commodities. So that is one of the issue there. So this is found at like 8 generation ending food loss in the global south, and this is some Arabic putting one from food is from our lab. So this is come by the food tank in the USA. This is the paper I would like to show that this is double layer coatings, means we started double layer one in order to achieve the both effect of the to reduce antibiotical properties as well as the quality controls. This is what we have done it. And these coating, this research, you can see that in the major journals become the top 25 hottest article in food chemistry, biology, different kinds of research. It shows that only 1, 4 or 5 articles. It shows that this is what the current scenario is. This is what the scientists around the world are looking into this area. This is what people around the world of food industry are looking into directions. That shows you the future directions, So this is you can see this came out 2020. This becomes also the top cited articles. Biology to again show you the quality of the work, or this is the future of the work. We need to work into that. This is many articles coming to that. I just want to. This is the paper actually. This is it comes in the special issues. Those people working around the world in this area, like Israel and ourselves from Malaysia and Luis Palau from Spain, working with DRS area. The chief editor asked us to put in the special editions, and this is becoming very popular articles. So you may go through that. This paper just come out yesterday. So this is strengths in nanotechnology, potentialities to control the plant pathogenic fungi just came yesterday. So if you want to know more about that, you can refer to these papers, So there's another one, it also come in like February 2021, critical comprehensive review of food science and food safety. So like somebody has mentioned earlier that we are wasting so much of food. Even in order to avoid, we can't avoid the food waste. What we can do that extract the valuable material from these. This is the current project. I got it from the UK, future food backend, and it's looking at protein from different dairy waste, from and animal waste, from like cereals, and from especially pulses. We are trying to add the value to that. So with that, I would like to thank all my collaborators who have helped me in one way or other way, and the great house we have managed some research. So this is my email, asker.ali@nottingham.edu. This is my lab. If you need to ask any queries or any other queries, I'm more than happy to collaborate. I'm more than happy to clear queries, other things. With this, I would like to thank Niftem again for giving the opportunity to talk. So there will be a question and answer session.
Yeah, we will take the question answers after last presentation.
Okay,
So,
I will refer with this. I would like to thank you
you
all
should
for this.
stay with us. Thank you. Yeah, so thank you very much, Professor Asgar. Your work is really unmatchable, and we all benefited from your presentation. And whenever you come to India, please visit our institute. We welcome you here. And
yeah, I would looking forward to have more collaboration in India.
Yeah, and also we want to collaborate with you in the post-harvest biology and technology. Earlier also we had some talk about this, and now we are in the position to start some projects as well as some centers here. So we want your advice forever to establish these centers here.
Okay, that will be great. Yeah.
Yeah. So thank you so much. Our next presenter is also ready. We are fortunate that Professor Patrick Webb is with us. Professor Patrick Webb has worked extensively on the food security policy and practice, nutrition, agricultural development, humanitarian emergencies, and climate change interactions with food systems. Today, he is the Alexander McFarland Professor at the Friedman School of Nutrition at Tufts University, US. He manages several large research projects, including Feed the Future's Food Systems for Nutrition Innovation Lab. Patrick also serves as the technical advisor to the Global Panel on Agriculture and Food Systems for Nutrition, advisor to the Food Systems for the Future Initiative, and serves as vice chair of the European Commission's high-level expert group mandate to assess the needs and design of an international platform for food system science, focused on climate change, food systems, and human health. Before joining Tufts, he was a member of the Science Council of the Consultative Group on International Agricultural Research, the CGIAR, and he has been a member of multiple expert groups supporting WHO, UNICEF, and FAO. Earlier, he lived in Africa while working for the International Food Policy Research Institute also. So I welcome Professor Patrick Webb in this webinar, and now we turn our presentations from technical to the food policy issues and how these advancement in the techniques are utilized in the food policies. So to tap on the future food systems, the growing need for innovations in nanobiotechnology, I welcome Professor Patrick Webb, and please start your presentation.
Dr. Sunil, and thank you to all the organizers for inviting me to this really fascinating and important meeting, and to all the speakers before me for sharing their insights. It's been quite instructive, I'm sure, for pretty much everyone here. So the reason I have this template, the Feed the Future USAID, is that the innovation lab that Dr. Sunil just mentioned, on food systems for nutrition, just started literally 3-4 weeks ago for the next 5 years, and is really focused on trying to identify promising technologies across the food system, not just in agriculture or processing, but all the way through, that can encourage not just less food loss and waste, but more access by everyone to affordable and healthy diets. And really where I'm focusing, we've been from the very nano to the very macro and back again. And fortunately, I'm the last speaker, so I'm really going to reinforce some of the things that have been said. But one of the key elements I want to bring to the table is the issue of changing a promising technology into a commercialized, scaled up product. We need to collectively find the ways to sell, both in a commercial sense, innovations so that they're cost effective at scale, but also sell the narrative, sell why this approach is important to the global policy community, and not simply something for scientists in labs to worry about. And I'm cognizant of time. I'm going to keep this pretty short and focus on the key elements. Now, when I say sell in a policy sense, one of the things that I feel that hasn't come out sufficiently today so far is that we need to fix the entire food system, not tinker at the margins and expand shelf life for 6 weeks or 8 weeks. That matters, but it matters in a wider context. And that wider context isn't just hunger. Let's not get focused just on the hunger index and India's decline in that over recent years. Healthy diets are at the core of all matters relating to planetary health and human health. And there's a big role to be played by innovations, including in nanobiotechnology, in improving both human health and planetary health. One of the big issues right now is that the food system, not just production, but all elements of the food system, contributes to somewhere between 1/4 and 1/3 of all greenhouse gas emissions globally right now. And that's a major issue, right? It's not the only issue, but it's a major contribution to GHGs. And we have to be cognizant of, in everything we do, including in research on biotechnology, is figure out how we can reduce the footprint of everything that we do in our food production, processing, storage, transformation, and transportation. But at the end of the day, what we choose to eat has huge implications for what is produced, how it's produced, and then its implications for planetary health. At the same time, those choices on what we choose to eat matter for planetary health in terms of human health. Diet, poor diet, suboptimal diet, not hunger, suboptimal diet, which means not good nutrient-rich foods, contributes about 11 million premature deaths every year. This is astonishing. And diet-related micronutrient deficiencies, certainly, hunger, certainly, but also non-communicable diseases are escalating across the globe with a disproportionate impact on low and middle income countries. There's no question that we know obesity, for example, is very high in countries of the rich north, but its non-communicable diseases are escalating fast. Diabetes in India, for example, less than 20 years from now, it's projected there'll be over 135 million people with diabetes. And the drain on healthcare systems and productivity, just of that, let alone stroke, high blood pressure, and so on. These are modifiable risk factors relating to diet, right? So what is eaten, what is actually available and affordable matters. So this means that we need to be thinking food system-wide. There's a lot of things that need to be shifted from a consumer perspective in terms of making the right foods desirable to a consumer. And I thought the example of millet being reformulated to look like rice was an interesting one. Affordability. We need to ensure that the nutrient-rich, dense, safe foods are affordable to everyone in every location around the world. That is part of the job of processing and transformation. Reducing the loss of nutrients, a lot of people have talked about that already, in the post-farm gate all the way downstream to the consumer. Huge role for biotechnology there. And then on-farm seed inputs, improved protection of products from pests and diseases. All of these things have to work together. And I would argue that young researchers need to be having the system-wide perspective in terms of where their work contributes to this wider picture to be able to sell, in inverted commas, their work. Now, there's a lot of actions needed in the public sector. There's no question, right? Governments still have a role to play in where they direct agriculture subsidies or research and development funding for public institutions. But there are many other domains that the public governments need to work in terms of leveraging, for example, public procurement systems to be more healthy. And India is leading the way in thinking around that. But social protection, trade policies, enhancing the science base for food systems choices, all of these matter. Because if they're all brought together, they're connected. Recent modeling has shown that healthier diets for everyone, this happens to be particularly a flexitarian diet that includes dairy and fish, are not necessarily going to be more costly to the consumer. This projection shows that actually by 2050, with reduced food loss and waste, fuller accounting of greenhouse gas emissions in the production side, and higher incomes and purchasing power, healthier diets could be less costly than they are today. So we have to keep that goal in mind, right? Right now, there's a mismatch between production and what are considered healthy diets. There's there are many ways that one can consider. There's no one diet that fits all. But right now, what everyone should be eating is definitely more fruits and veg, and we've heard about the innovations in that space. Right now, it's mostly starches and sugars and oils and fats that dominate. So we have to find ways to make those nutrient-rich foods more available. I'm going to skip this in the interest of time. How do we think about these greenhouse gas emissions? Well, by 2050, animal source foods are clearly the major contributor, and that will grow. But it's not only there. Staples are a major user of so-called blue water, unused prior. Fruits and vegetables, major contributor to overuse of nitrogen applications, and other crops, whether they're seeds and nuts and others, phosphorus applications. We have to think in our nanobiotechnology domains, how can we reduce the requirement of application of resources, right? That's one way. Another dimension is to focus on the seeds themselves. How can we potentially reduce phytates, which are a binder? They're an anti-nutrient. They make nutrients that have been put into seeds less bioavailable as a food for humans. But there is research that shows that lower phytate grains, for example, can be produced without impairing the growth and productivity of the crop. Downstream from the farm gate, we've heard a lot about food loss and waste and the huge potential. This is a major contributor. Almost 10% of greenhouse gas emissions relate to food loss and waste, partly through emissions as a result of that, partly through the fact that you have to produce it again once it hasn't reached the consumer. This is crazy. And in many parts of South and Southeast Asia, much of the food loss and waste is happening in that value chain space between production and consumption, in the transformation and packaging of foods. So we need to keep that picture very much in mind. Now, there's a huge push these days. I was involved with the World Economic Forum, which was very much looking at what are innovation, where are those innovations? And many of them are considering the role of nanobiotechnology and other kinds of technology, be it blockchain or the Internet of Things and so on, to bring together, to allow for this connectedness of sensors, innovations in sensors, moisture meters, everything that relates to biofilms and beyond, all of which can reduce that food loss and waste. In terms of large numbers, millions of tons, may not look like a whole lot in terms of percent of total food losses, but this is early days, and most of these innovations have been applied so far in high-income countries. The question is how to make them affordable in low-income countries. Many of you probably have seen this recent review of the innovations that are out there, innovation potentials that go from research in the lab or on the field through validation to prototyping and then implementation at scale. And there are a lot of these things that have been identified, for example, biodegradable coatings and food safety technologies and sensors that are already in the prototype domain. Much of the focus in this assessment, words that have the word nano in them, are considered to be somewhere between research, discovery, and validation, experimental proof of practice at large scale. That is something we need to accelerate. We need to bring all these things with the word nano faster along this chain to use, adoption, and commercialization. Quickly, I'm just going to say that this doesn't relate to nano, obviously, but my point here is about selling in terms of commercialization. There are innovations in other spaces for improving the rapidity of drying, for example, using solar dryers in Bangladesh that draw hot air through shelves of fish or fruits and vegetables that are being sliced, dries them more quickly, which brings up value added, but also protects them from flies and dust and the rest. One of these costs $120. That's actually quite expensive US dollars in a local context. Sorry. But the real point is that we have to understand that people use these technologies not always in ways that are predictable. Now, what this shows is the use of a chimney dryer in 3 different locations of southwest Bangladesh. And what they did, the farmers were encouraged to use this for fruits and vegetables. They used it for all kinds of other things. And the taka, the amount they generated in terms of profit, net income, differed considerably based on how those farmers actually used this and how much of that produce was sent to market, the commercial dimension, not taking even into account the startup cost, the cost upfront of investing in the risk-taking involved in technology adoption, which can be quite considerable. So we have to consider cost effectiveness from the farmer's perspective, the commercial perspective, from the very beginning. This just offers an overview from a recent publication from India on all the domains in which nanotechnology can be applied across the food sector. And I totally agree with all of these. But the key is potential and innovation. We can't stop with those words. We have to start with those words, but they have to be moved more quickly to the commercial domain. This shows globally where all crop and livestock and seafood production is produced, the bulk of it in the world. And of course, India is right up there, which means that there is huge potential for these innovations, not just to be made as potential solutions to these planetary health and human problems, but actual solutions. So we know the planet and the people on the planet are hurting. There's no question. But let's focus on the positive. There are growing numbers of solutions, and these innovations that we've been hearing about today so far have huge contribution to play, huge potential. One thing that hasn't been talked about much is the opportunity for income growth, for companies, for young people, for entrepreneurs to get involved in this space, not just from the science side, but from the commercial side. Job growth matters hugely going into the future, and this is a space in which that job growth will play a huge role. But the two encouragements I would put on the table is, one, make sure that you build in net cost benefit calculations into the research from the outset. And what are the mechanisms by which innovations can lead to commercialization? And I don't just mean through conventional conduits, through ministries of agriculture or extension services. What are the mechanisms for dissemination that could be built in to prioritizing the research and encouraging that research to be upscaled? And the second one is not just to focus on the issue of hunger and nutrients and so on, but consider how and cost out how such innovations could actually help reduce the planetary impacts of the food system. Cutting natural resource use, reducing greenhouse gas emissions by engaging in innovations in nanobiotechnology that can help sell this space to policymakers who are increasingly concerned of not just about producing affordable, healthy diets, but creating jobs and reducing planetary impacts together. So I'll stop there. I thank you very much for inviting me and look forward to engaging with all of you in the future. Thank you.
Thank you so much, Patrick. You have given an overview on the future food systems and how we can incorporate nanobiotechnological advances into that. So we are very thankful to you. I invite Dr. Neetu Taneja for taking some questions. So if there are some questions, directly she can take, or otherwise raise the hands, and after that she can take the question. So Dr. Neetu, please coordinate for the question answer session.
Sure, sir. Thank you so much. Thank you to all the speakers and very, very talk this biology. And there are a few. The first one is there for Dr. Ali as well, where a participant wants to know that what is the most suitable way for easy drying of chitosan coating, since it is very viscous in nature.
Sorry, can you repeat again, please?
Yes, sir.
What are
Yes, sir. So basically, since chitosan is very viscous in nature, the participant wants to know what is the most suitable method of drying, a suitable and easy method of drying after the chitosan coating has been made on the surface of the fruit and vegetable.
Okay.
Due to its viscosity. Yes,
yeah.
Sir.
Yeah, I think the best way is to we leave it into the ambient temperature and using fan. You know, and depending how you dissolve, that is going to affect the viscosity of this chitosan solution. So normally we use we tried different kind of organic acids like oxalic acids and acetic acid and malic acid. What we have found that acetic acid 0.03% is the best one in dissolving this chitosan. So we started, we have like high molecular weight, we have a very high viscous solution. And after that, what we found that even low molecular weight is much better than the high molecular weight chitosan. There were several problems when you're dealing with the high molecular weight chitosan because of highly viscous in nature. When it formed the coating on the fruit surface, it cracked. So when we use the low chitosan, that there was not much problem with that. So I wouldn't recommend to use the high molecular weight chitosan, And when you look at the antimicrobial properties, the low molecular weight chitosan has a better efficacy as compared to the high molecular weight of this chitosan. And easy to dry, easy to manage.
Okay, sir. Right. Thank you so much. There is one more question for you, sir. In edible coatings, the moisture of the coating material has to be dried before the transit of produce in the supply chain. And for commercial scale up, apart from air drying, how would it be possible to achieve the same fate without compromising the quality of the produce? So in a short span of time, what would be the application, or what would be the method for this drying, sir? Especially when the produce is in a very, like it's there in high quantities, in tons.
Okay, so I think the best simple one is like we use like dipping methods. We tried different kinds of method like spray. We use spray to coat the fruit, but ultimately what we have found that dipping is the best way, because sometimes the morphology of the fruits matters a lot. Like dragon fruit, if you spray, you can leave something like on the edges and the end scale or something like that. But in the dipping one is the best one. So we use like a mechanical strainer. We dip the fruit for 1 minute and then use the strain and leave it like that. No, so dipping is the best way compared to spraying and any other method what we are brushing or spraying.
So, sir, would it be dependent on the time interval, how long the dipping is being done? The dip coating, or is it just one soak, or several times the method is repeated?
No, it's one way, one time only. And also, what is your effect? Whether you are looking for like look at the antimicrobial properties or look at the something like post-harvest quality or shelf life. It depends on that. Like when we use like nano emulsion one, it's very short duration is fine. When we use the conventional one, then you have to leave it for like we found that about 1 minute is the best one as compared to other one, because it also needs some time to penetrate, because this chitos has an induced resistance phenomena. So it has to reach to the cells. So you have to dip it for some duration, in order to have a best efficacy. So 1 minute was the good one.
Okay, sir. Right, sir. Thank you so very much. There is one more question, and I think it is for Dr. Patrick. Is there any way to reduce the carbon footprint of foods, Dr. Patrick?
Of foods or fruits?
Of foods in general, sir.
Of foods, yes. So, yes, absolutely. One often sees graphics that say meat production is so much more, generates more methane and other types of greenhouse gases than legumes, than chickens, right? But in fact, the technology, the approach used in producing various crops can vary considerably, both in terms of the amount of water, the amounts and types of inputs, including fertilizers and pesticides and so on, as well as how intensive versus how extensive the production is, right? So it's not just what is produced, it's how it's produced as well. And so we need to focus on higher quality inputs, seeds, as well as livestock health, as well as other things, to increase the efficiency of productivity, because that reduces pests and diseases, which in turn reduces the need for pesticides, herbicides, and so on. So we need to think of it in that more holistic sense. And nanotechnology can certainly play a role in that space.
Okay, sir. Thank you so much. For Ali Asghar, sir, there are a few more questions. Any other biomaterial which is helpful other than chitosan? Is there any other natural polymer which you suggest would be a good alternative for nano emulsion, sir?
Correct.
So besides chitosan, are there any other biomaterials which would behave the same way as you have suggested?
Yes, I wanted to show you, but due to the lack of time, we have been working on that. There is another coating we developed from gum arabic. So if you look at the gum arabic, it is like a gond. We call it gond in India. So that is very famous, and that is how it comes out. This is a good emulsifier being used in Coke, Pepsi, and inks and other things as the emulsifying agent. I was talking to one guy from Sudan, because Sudan is a major company producing gum arabic. So this become more popular than the chitosan itself, just because of it's a vegetarian and it is coming from the plant sources, and this has a similar effect as a chitosan. What is missing here is microbial properties, because this gum does not have any microbial properties. So what we did, we can use very small, very minute amount of chitosan. If there is a problem with the non-veg, then we use like propolis. I wanted to show you the propolis, which is very high in phenolic content. And this is coming from the beehives. When take out the honey, the remaining portion, you can extract it, it's phenolic extract, and that is a very good amount of high phenolic content, you can find it. So we encapsulated this one into the gum arabic based coating. So that is suitable for the vegetarian people. It has become more popular as compared to the chitosan one.
Right, sir. Okay. So gum arabic is one of the best alternatives. And what about carrageenan? I mean, and sir, what about carrageenan? Because there are a lot of polymers which biopolymers which people have shown. So carrageenan, what is your opinion on its application?
I think
Is it used as a coating or an emulsion, sir?
it can be used. Carrageenan is becoming famous to some extent. I didn't work much on this carrageenan, but there are some other materials. You look at the properties of these edible coatings. Some are like hydrophobic and some are hydrophilic. So you need to look into that. So some coatings coming from polymers, some people coming from lipids and different. So they have different properties. Like so the one is coming from lipids and oils. This is like a hydrophobic. The polymer, the coming from like chitosan, this is something like is not very much hydrophobic. So you can blend into, so what in my center, what we are doing, blending, making it into a composite one to get the better effect. So I would say that making composite coating is a better way forward. So you can get like mixing this lipid-based coating into the carbohydrate-based coating or something like that.
Okay, sir. Right. And just one more query from one of the participants. When you said that the dip coating is one of the best method which can be actually used on a large commercial scale, would simple air drying or use of fans be sufficient for it, or any other kind of technological intervention would be required for completely drying the coating?
I think ambient temperature is very much suitable. I think we just look at the ambient temperature with the air con. Sometimes just leave it 3-4 hours is the best way. Otherwise, if you use the forced air drying, that might have hurt the coatings, and maybe it can tear off from the surface. So what we found that like simple air drying is the best way. Of course, if you're using some nice temperature like 20, 20, 25, something like that, that is the best way.
Okay, sir. Right. Thank you. Well, I don't see any more questions in the chat box. So if there are any further questions, the participants might just put it in their chat box. Okay, there is one more comment from one of the participants, Mr. Sahil, and he's saying that there is a trend of integrating essential oils with chitosan nano emulsions. Yeah, so but this, does it actually reduce the sensory property of the food by incorporation of the essential oils?
Good.
Does it reduce?
This is a very good question, and I showed you 1 or 2 slides on that, right? We are also incorporating essential oil because these are very good material in terms of antimicrobial properties, And when you use this, what is called these oils, you need to use in a very, very less amount, or we just encapsulate into the main edible coatings. So sometimes, and we have done the sensory analysis, it is not that much, like banana is very much fine, like dragon fruit, it is maybe apple or something, it may affect the sensory quality, but depending how much we are using it. If you are using complete essential oil, that might have problem. But we are incorporating very, very low concentration into the, and when we use, you can see when we use the what is called essential oil, there is a phytotoxic effect. One slide on the dragon fruit, when we use essential oil, there is a strong phytotoxic effect. When combined with gum arabic, then effect was reduced. So I wouldn't suggest that you use completely essential oil. I think it's no use. These are very expensive, and a small amount combined with some other edible materials, then you use it. We didn't see any problem with the sensorial analysis.
Right, sir.
Trying on chicken fillets nowadays, yes. This recent paper came out this week. So we are trying on the chicken fillet also.
Okay. Right. Okay, thank you so much, sir. And there is another question. One question is there for Dr. Subramaniam. And sir, the participant wants to know the mode of action of hexanol in increasing the shelf life of the produce.
Yeah, as I mentioned, hexanol is a natural compound. It's a wound responding compound. Whenever you bite a cucumber or any fruit, this compound is naturally produced. It is by chemistry, it is hexanaldehyde. So what we have done is we put it in a nano emulsion and spray on the fruit. There are two reactions going on after the spray is done. The first reaction is it entered into the fruit and reacts to the phospholipase D, which is localized in the middle lamella of the fruit cell wall. So that is slowed down because of the hexanal. The secondly, it also reacts on the ethylene pathway. It slows down the ethylene evolution from the fruit. So those are the two reactions going on after the spray is done. After the sprays, we also monitored whether hexanol get transformed into the fruit. Within 24 hours, we will not be able to detect hexanol at all because it is metabolized in the fruit. And whatever the reaction happening immediately after the spray or within 24 hours, the effect stays for about 3 to 4 weeks. And that's why we have done lots of work. And being a 12 crore project, we have done 40 theses on this particular formulation on several crops, several fruits.
Okay, sir, that's really nice. And there is another question for the panel. Basically, the participant wants to know that what could be the major thrust area of research for postgraduate students in the area of food packaging, especially when we are talking about biopolymers as packaging materials. So what could be the most vital thrust area of research as far as PG research is concerned?
I think for packaging, I mentioned about the nanocellulose, which is derived from the plant-derived cellulose, because we have lots of huge waste. Banana bunch is harvested, you get a pseudostem. You get agave americana, it's a fibrous plant. Pineapple, after the juice is extracted, you get fiber. You get lots of cellulosic waste. So that can be converted into biodegradable film, which is one of the immediate and potential area. Two of my PhD students working on that, and I have published 3 or 4 papers in bioresource. You can refer to that, and that is one of the areas. And right now the price is high, but if you could make it commercially at the large scale, the price will go down and it will be adaptable. So that is the immediate thing required for India. Even such films, we also made it from the wood waste. What were the timbers after seasoning a wood or making furniture, you get waste. So that can also be converted into cellulose. Basically, cellulosic waste converted into biodegradable film, which is a very potential area for research. That is number one. Number two is encapsulation of functional foods. And particularly India, you have varieties of fruits, varieties of vegetables, which rich in metabolites, rich in biomolecules, active, antioxidant compound and all. So if you do the encapsulation, which can be done in a master's thesis, it is an excellent work. Do some, research and encapsulate it successfully, and do some characterization. It could be a good thesis as well. It will lead to product development.
Right, sir. Thank you so very much. And there is one last question we'll take in the interest of time. That is, what is the recommended limit of hexanol to be used in fresh produce, as per the Government of India guidelines?
Okay, Government of India, actually this is developed involving Gopi Palliath in University of Guelph in Canada. The recommended level is 2%. 2% is a diluted form. We have, 1 liter is dissolved in 50 liters. So a trace of hexanol is used. For every liter, it is only 1 ml or 1 ml of hexanol. It's a very limited quantity. It's insoluble in water. That's why we put it in a nano emulsion to make it soluble and enter in the fruit, so on. So basically, it is a very friendly chemical. Already USDA recommended this particular hexanol formulation as a food additive. Suppose we want to keep the greens fresh, and this can be sprinkled on the food. Already it is FDA recommended product only. And the Canadians call as a feed additive for the cattle to feed on the grasses. So this is sprayed. It's a grass compound, generally regarded as safe. Also a grass odor compound. When you mow a lawn, you get a odor that is a grassy odor. That is chemically referred as hexanol. It's a very safe compound. Nothing will happen. So we are all safe and we have eaten enough fruit which are treated with hexanol.
Right, sir. Thank you very much. And thank you all. There are no further questions in the chat box, and I think we are nearing the conclusion of this webinar also. So I would like to invite Dr. Chindi Vasudevappa, sir, Vice Chancellor Niftem, for the concluding remarks, please. Thank you.
Yeah, good evening, everybody. And I would say this was an excellent session we had, probably for the last 3.5 hours. I think I would say it actually exposed all aspects or facets of, I would say, nanobiotechnology. That was really great. And I wish to thank each one of the speakers for their eminence and expert talks. That's something which is really great, and all of us are very benefited. I would say now my sincere thanks to especially Dr. T.R. Sharma, the DDG, Soil Science from ICR, and he stressed on one important area, maybe the phytotoxicity and lycosome damage, and maybe the lack of toxicological information. These three things are very, very important, I know, because probably Dr. Patrick agrees with this, because these are all the information which are essentially required to promote nanobiotechnology. Otherwise, as he said, of course, it is a very big area where he touched upon, especially the food systems approach and how to reduce the carbon emission and things like that. It was really great. And probably as agricultural scientists, we need to look back and see how best we can bring in the concepts of nanobiotechnology to reduce these things. I think that was really great. And of course, Siddharth Thiwari was as usual, because I have heard him 2-3 times. And in fact, Nabi has been doing wonderful work, both basic and applied. And the golden banana, I think they are taking forward in a very big way. And already a lot of patents have been developed, and probably including, I would say, the packaging, they are into it, and even the food products. And I am happy to mention here that we are collaborating with them. And probably I wish we collaborate with all the other labs. Today, in fact, I was very happy to see Dr. Subramaniam giving a lot of insights into applicability of the whole nanobiotechnology, especially the coatings, the emulsions, the polymers, everything. I think it was really good. I would say that students will be very much benefited. And so also even the budding scientists who want to really get into nanobiotechnology. That is something really great. And thank you very much, especially Dr. Subramaniam, for your excellent, I would say, talk. It was wonderful. It was really an eye-opener, because you touched upon various, I would say, fruit products of India, the fruits, vegetables, and all that. Of course, I would not to belittle Professor Asghar Ali, and he was very good and maybe an authority, especially on chitosans. I think that is something really great, and he was giving a lot of insights into the utilization of chitosans. One thing which may really recur in India, countries like India, is chitosan is derived from crustaceans, which probably people may not really like as vegetarians. That is one thing which always the taboo will carry forward. I think that is one thing probably you can address, even the alternatives also you have given. And that is something really, and I would say, very great. And your lab is doing a lot of work. Probably it is an authority on chitosans, I would say that. And excellent work. And probably I wish Niftem, as a food institute, we wish to collaborate with you. And since you are our own person and working for the university, Nottingham University in Malaysia, probably that's a great, and I would say it's a great moment. And of course, we had very excellent, I would say, eminent talk, especially from Patrick. I think it was really wonderful. And it was more on a policy perspective, and how do we take it forward as a global entity. That is something really great, and we need to really look at this very seriously, because I know we have been talking about the 10 billion population and the enhancement in food production and the converting the food losses into various other products and things like that. At the same time, how does the nanobiotechnology reduce the CHG, GHG? That is what is very important. Probably that is one area. In fact, I wish to just make a mention here. The Food Future Foundation has been started in India. Probably you must be aware of it. And UN is also involved in it. And of course, we are all members of it. And I would say that is one good area where a lot of things can be done. And probably I wish you can give the directions for the future research in nanobiotechnology. That's where I feel it was really very, very important and wonderful. I think, today we had, I would say, a wonderful talk from each and every one of you. And not to forget, especially, of course, the contributions from our own institute. Especially, I would say, Dr. Parikh has done a wonderful job. He has brought in eminent people onto the panel. That was really great. And I know it is not easy, but he has done it in very short time. And I wish to convey my thanks to each one of you for accepting such a invitation within a very short time and making you available for presenting your views. Wonderful. And I would even further say, even the delegates, in fact, they were about 120 in the beginning. Now they are about 100. That means people have not gone out of the sessions. That means the sessions were so very interesting. And this is a new phase of science. Probably we need to look forward for a better research innovation and probably application of it in the larger interest of the society, and also, of course, meeting the food and nutritional requirement of the people. So with this, once again, my pronouns to everybody from India, especially for Patrick and Askar, and also for all my friends, Dr. Subramaniam and others. And thank you very much for your, I would say, kind involvement. And I would say the interesting sort of presentations which you all made. I think it was something really great. And I wish we will have you once again. Maybe whenever you come to Delhi, probably don't forget to at least call us, and we will be very happy to have you here, and probably for a day and all that. And I will be very happy to even interact. And I wish our faculty will interact with all of you and try to get some input so that we can initiate, especially more focused on food. So that's what I wish to say finally as a concluding remark. Thank you very much once again. All the very best. Thank you. Thank you.
Thank you very much, Vice Chancellor and others.
Thank you,
also.
sir. Yeah.
Thank you. It's a great
Thank you,
event.
sir.
Thank you very much.
Thank you. Thank you.
Thank you, sir.
Thank you very much. Yeah.
So thank you all, and Parikh sir, we can conclude.
Yeah, Dr. Netu. My job has been done by Vice Chancellor itself. He has given thanks to all our speakers and the participants also. I see here that many of the participants, to them only in the night 11 o'clock I sent the mail, especially to Professor Maria Turtoy from the Romania, Isliya Yildiram from Turkey, or my friend Professor Muhammad Aziz from Morocco, Ramesh Deshpande from International Agriculture Advancement Group. We have met it with Professor R.S. Parodaji, and he is at the Washington DC. Earlier he was with the World Bank also. So all the eminent persons have also joined. I also thanks to one and all the speakers, and my sincere thanks to our Vice Chancellor, Professor Vasudeva Paji, who has given us the chance to organize this webinar. And also he stayed here for almost 4 hours full time. He is here, and his involvement is seen in his concluding remarks that he has not even miss a single slide, and he has given the remarks on all the presentations here. So we thanks to all, and I also request to all the presenters that they should send the salient points for making the proceedings, and we will send these proceedings to the government also, so that something can be out from this webinar. I really thanks to all from the core of my heart.
Thank you once again. Thank you. Good night. All the best. Of course, for Patrick, good morning. Wish you all. Thank you very much.